How did the primordial, hell-like Earth develop into the cool, wet place we call home? What range of conditions can different forms of terrestrial life survive? We'll delve into the fascinating world of hardy cells that thrive in boiling acid or pockets of saturated salt water trapped in solid ice. All the life within the ecosphere ultimately depends on light-catching algae. We shall marvel at entire communities that survive in the dark depths of the oceans and bacteria miles underground, which eat rock and are completely independent of the sun's energy. Could similar cells exist in the solar system's niches? We will explore other prime astrobiological locations in our backyard, visit liquid water aquifers deep beneath Mars' rusty surface, float among the clouds high in Venus's atmosphere, and drift through the frigid global ocean trapped under Europa's face. What about worlds even further afield, orbiting the pinprick stars scattered across the heavens? Astronomers are discovering new planets at a prodigious rate. Which of these are the most likely to be inhabited? What is a red dwarf? Could photosynthetic organisms be soaking up the rays of a dim red alien star? Might cells survive even in outer space? Could life, in forms we can only begin to imagine, spread like a cosmic infection between our solar system's planets, or even between stars? The potential for life beyond our Earth is a concept that ignites the imagination and inspires wonder. The field of astrobiology is a rich tapestry of diverse disciplines. It involves the study of diverse extreme organisms on Earth, research into the origins of life on our planet, and discussions about the necessary processes and environmental conditions. It is the search for potential niches for life, both within our solar system and throughout the galaxy. It is the design of lab experiments into prebiotic chemistry, or alternatives to the idiosyncratic systems developed by terrestrial life. It is the construction of rigorous life-detecting experiments and instruments flown on space probes throughout the solar system. This interdisciplinary nature of astrobiology adds depth and complexity to the field, making it a fascinating area of study. Astrobiology is multidisciplinary, and it is difficult to define the field of knowledge it covers. As expected from a nascent discipline, some aspects rely on speculative or incomplete data. Some critics have described it as a science that has yet to demonstrate that its subject matter even exists, in that no single example of life beyond Earth is known. This is undeniably true, but not in the sense of doggedly searching for a mystical beast like the Loch Ness Monster. In science, we postulate hypotheses, construct experiments, refine theories based on data, and design improved experiments. Astrobiology was not born to explain a specific discovery, just as microbiology developed to explain sightings through the first microscopes. Instead, it is the product of more mature scientific disciplines that overlap. Astrobiology can point to no specific inception, but has gradually gathered momentum and acceptance over the last 50 years, as discoveries within biology and space exploration show that extraterrestrial life is possible. I shall begin the story with the most fundamental question. What is life? What is life? This seemingly straightforward question has plagued biologists, philosophers, and theologians for centuries. It is mindlessly simple to discriminate between what is alive and dead. Jaguars, oaks, and mushrooms are obviously living. Various time or size scales exist. Over an extended duration, one can observe the growth and development of lichen and the colorful crusts on old stone walls. Chemical analysis of the surrounding air confirms their photosynthetic activity. Magnified 50 times through a microscope, a drop of pond water teems with minuscule organisms. On the other hand, rocks, fires, and clouds are clearly non-living. However, simply providing a list of living things is different from being able to concisely define the properties of life. School children often learn that a checklist of 7 characteristics defines life. Living things eat, excrete, move, grow, reproduce, respond to environmental changes, and maintain a constant internal state. Some non-living entities satisfy a few of these attributes. Fires grow and spread, self-sustaining from a flow of energy, consuming fuel and excreting waste products by the same oxidation reactions that run a cell, and the ordered pattern of atoms in a crystal can reproduce itself. Conversely, some living entities do not meet all the criteria. Mules are sterile and incapable of reproducing. The entire animal fails the 7-part test, despite each individual cell being alive. We need a more sophisticated definition. The Darwinian definition, which aims to characterize existence, is one attempt at defining what life is. It is an attempt at characterizing life. First, it states that life must contain a description of itself, an operating manual, or instructions on rebuilding it. Thus we exclude crystals because they lack a true self-description and only grow because their structure arranges free units into an existing pattern. Second, it states that the individual must be able to carry out the instructions independently and self-replicate. This rules out viruses, as they reproduce by hijacking the molecular machinery of the host cell they have infected. All life on Earth has its own operating manual, a text, a set of genes within the DNA molecule. A vast array of proteins interpret and carry out the instructions. Therefore, this classification type also goes by genetic definition. The term genetic here refers to a much broader concept than just a section of DNA. It is closer to its Greek root, meaning mode of formation. Third, it states that the system must be able to evolve through natural selection. This suggests that the process of replicating genetic information should be imprecise, leading to the introduction of errors or mutations. This introduces random variation within a population of replicators, ensuring that only a few survive to reproduce in the face of environmental stress. This is the mechanism of Darwinian evolution. Over time, the replicators adapt to become better suited to their surroundings. This process honed the abilities of the first replicating molecules to produce cells and animals, and eventually, almost 4 billion years later, the self-aware species we are. People often assert that the human body is an intricate organic machine created exclusively to facilitate DNA replication. The Darwinian definition prescribes that life needs only a system of information storage and transference, transmitting the genetic instructions to the next generation. What defines life is its actions, not its composition. This classification is much less restrictive than others and includes non-biological life. Artificial or A-life development is a burgeoning field with many different systems, such as replicating computer code and replacing organic polymers and hard disks as the primordial soup. The processes of mutation, competition, death, and evolution are the same, but only the supporting medium is different. A second definition of life specifies that the system must extract energy to maintain itself, in addition to information transmission. The problem is that a self-replicating system demands an extraordinary level of complexity. Complex organizations are improbable. There are vastly more disordered ways to arrange a cloud of atoms than ordered into a functioning cell. Everything in the universe naturally deteriorates from high order to greater disarray. In technical terms, systems fall down the entropic gradient from an ordered state of low entropy to an equilibrium level with greater entropy. Life constantly fights this trend towards degeneration, keeping itself far from equilibrium. It does this by pumping in energy. Maintaining an ordered state takes work. One can extract energy from something as it degenerates. For instance, an ordered wooden log emits heat when a rush of oxidation reactions reduces it to ash and hot gas. Life effectively enables one system to slide down the organizational slope, pushing another system uphill. A fungus that sprouts from a tree stump survives by extracting the same energy as a fire, but in a carefully controlled manner. Life requires a constant energy flow and can only survive where there is an external gradient. Later in this chapter, we will see how different terrestrial life forms extract energy from their surroundings. Modern terrestrial life performs these functions elegantly. It holds a complete description of itself and an elaborate network of chemical reactions that release energy, harnessing it to build useful molecules and maintain its complexity. An army of proteins oversees this metabolic network, providing the machinery to carry out the instructions in the DNA and copy them for the next generation. An enclosed space serves as a third attribute of terrestrial life. All life on Earth is cellular, bound by a membrane that physically separates the inside from the outside, preventing the different components from simply drifting apart, allowing control over the internal situation, the import and hoarding of valuable nutrients, the exclusion of waste products, and the creation of chemical gradients to allow energy generation. Traditionally, scientists believed that our planet had only two fundamentally different life forms: eukaryotes, including our animal cells, plants and fungi, store their DNA in a nucleus. Bacteria represent another more ancient form of life without a nucleus, called prokaryotic, meaning before the nucleus. Their DNA, twisted into a closed loop, floats free within the cell's watery cytoplasm. This is one of many differences between the two cell types. Many smaller parts, or organelles, divide the inside of a eukaryote. These include mitochondria, the powerhouses of the cell that perform numerous reactions to extract energy from food compounds and produce adenosine triphosphate, ATP, an energy-storing molecule, and chloroplasts, found in plant and algae cells that carry out the light-harvesting reactions of photosynthesis. The process of protein synthesis is also much more sophisticated in eukaryotes. Like their prokaryotic counterparts, eukaryotes concentrate their protein-making ribosomes on a unique membrane extending from the nucleus. New proteins pass to a stack of organelles and undergo step-by-step processing into a finished product. Thick protein strands strengthen the outer membrane of eukaryotic cells and form long poles that can drag other proteins or entire organelles. These protein filaments allow for a much more refined cell division than prokaryotes. They also give the eukaryotic cell superb control over its outer membrane, which it can use to crawl over the surface or predate on smaller cells to engulf and digest them. This final ability explains how eukaryotic cells acquire their mitochondria and chloroplasts. Both of these organelles are believed to have once been free-living bacteria. The evidence is abundant. Mitochondria and chloroplasts exhibit numerous characteristics of bacteria, including circular DNA strands and antibiotic sensitivity, and they do not originate from the host cell but self-replicate. An early eukaryotic cell eventually swallowed them, but did not break them down. Throughout evolution, the bacterium and its host cell became so interdependent that they are now quite inseparable. The nucleated cell depends on the energy provided by its mitochondria and chloroplasts for photosynthetic eukaryotes. In return, the organelles receive essential nutrients and protection. Symbiosis refers to this close partnership, while endosymbiosis refers to the coexistence of two organisms. Eukaryotic cells are generally much larger than prokaryotic cells to contain this internal organization. The total number of prokaryotic cells living on and in the human body outnumbers the amount of eukaryotic cells by at least 10 times. You're more bacterial than you are human. There is an enormous difference in scale between the smallest bacteria and the largest eukaryote. This traditional understanding that there were two fundamental life forms, cells with a nucleus and cells without, was the story until recently. Distinctive features such as skeleton traits trace the evolutionary history of mammals through the fossil record. But similar techniques are impossible for the minute features of single cells. However, in the early 1980s, scientists used a new technique based on the letter sequence of a specific gene to draw the family tree of all life on Earth. This gene codes for a small ribosome subunit, the tiny structure that translates the DNA code into proteins. Because of this process's fundamental nature, all organisms share parts of the ribosome gene, allowing for the calculation of an organism's closest relatives based on sequence changes between different gene versions. The results were astounding. As expected, the eukaryotes, the cells with a nucleus, cluster on one enormous branch of the tree of life. Surprisingly, prokaryotes do not form a single group, but divide into two major domains, bacteria and archaea. The genetic differences between human cells and the bacteria that cause stomach upset are as significant as those between an archaea living in a hot spring. This wonderful tree's root, which serves as the hub for all three bushy domains, is also fascinating. It reveals the closest living cells to the universal ancestor, the organism from which all life originates. The true tree of life is much messier than the one commonly depicted. Throughout evolution, cells frequently exchanged genes, which significantly complicated their relationship. Furthermore, the evolution of eukaryotes involved the wholesale importation of entire bacteria to form mitochondria and chloroplasts. It is uncertain whether the original host cell was a bacteria or archaea, but extensive gene studies show that eukaryotes contain far more bacteria-like material than archaea. The eukaryotes connect the two other domains, transforming the tree into more of a ring of life. We can summarize a cell as a membrane-bound chemical reactor and information storage system. But how is matter animated to produce life? What molecules are responsible for these amazing abilities? The three main components of a cell are the outer membrane, the genetic system, and the metabolism. A layer of molecules, with head ends that dissolve easily in water and fatty tail regions that do not, forms the cell membrane. Arrays of these molecules spontaneously arrange themselves to keep their fatty tails out of the water, forming a double layer with the tails aligned end to end in the middle. Metabolism is a staggeringly complex network of reactions that interconvert cell chemicals. Chemists believe that many small organic molecules, defined as particles containing carbon, originated naturally from non-biotic processes on the early Earth and integrated into metabolism during its development and expansion. Over time, life began to invent new, larger molecules to serve its functions. For example, long chains of sugar subunits, such as starch, are a compact way of stockpiling energy and carbon. Arthropods, such as insects and crabs, use chitin to construct their rigid outer skeletons, and a wall of amino sugar polymers protects bacteria. Cotton shirts, wooden bookshelves, and the pages of this book are all mostly cellulose fibers, a plant polymer built of glucose units. However, proteins are the ultimate polymers because of the sheer diversity of structures and roles within the cell. Proteins serve various functions, such as providing structural support, transporting valuable chemicals, transmitting signals, and acting as enzymes to accelerate metabolic reactions. The subunits of proteins are amino acids, a class of small molecules with acid and alkaline ends, and a side group that gives each amino acid slightly different properties. 20 amino acid combinations construct all proteins on Earth, with only a few exceptions. Some side groups dissolve well in water, while others are insoluble. For a protein to be soluble, its long chain must curl into a complicated three-dimensional shape, held together by bonds between the side groups, protecting the insoluble amino acids inside. The finished shape is extremely precise. Proteins can recognize and bind to other proteins, DNA, RNA, or metabolic reactants, with exquisite specificity. One class of proteins, enzymes, are custom-made tools that catalyze reactions, enable the chemical processes to occur much more rapidly than they would otherwise. Each enzyme can only make one small change to a compound, perhaps sticking on a phosphate group, removing a hydroxyl unit, or breaking a bond to snap a carbon chain in two. complete metabolic pathways contain many enzymes that gradually transform one molecule into another. The stability of enzymes and other soluble proteins is a key issue in biology. Warming enzymes cause the amino acid strands to vibrate. At high temperatures, this vibration breaks the bonds between side groups, causing the protein to lose its vital shape. If water also enters and exits the solution, it destroys the entire protein structure. Denaturing occurs when you fry an egg, causing the albumin protein to solidify and turn white. Denature a protein by changing the electrical charge around it, such as in extreme salinity or acidity. This is similar to the process of marinating raw fish in lemon juice. In the following chapter, we'll look at organisms that thrive in extreme environments, such as boiling hot springs, saturated salt solutions, or highly acidic water, and see their evolved coping strategies. Another important aspect of the molecules is that cells use their handedness. Any non-symmetrical shape possesses a quality called chirality. To see it for yourself, put down this book for a second and hold out your two hands, palm side up. Although your hands are identical shapes, orienting one onto the other is impossible. They are mirror images. No amount of rotating or flipping your right hand can make it look like your left hand. The same is true of most molecules in a cell. They have a particular handedness or chirality. Enantiomers, the two possible mirror image versions of a molecule, differ as much as your left and right hands. Intriguingly, all terrestrial life uses just one enantiomer. All biologically produced amino acids are left-handed, whereas all sugars are right-handed. However, the molecules produced in labs that simulate the early Earth's prebiotic chemistry are always an even mixture of both enantiomers. It is simple to understand why life only bothers with one enantiomer. The enzymes running metabolism are so specific that they can only work on their target molecule if it is the correct chirality. Life can only tolerate one enantiomer because having both left and right-handed versions of each enzyme would be extremely wasteful. What initially led to selecting one enantiomer over the other remains a mystery. According to astrobiologists, the presence of one enantiomer is a powerful biosignature of alien life. We created instruments for the Mars probes to test the chirality of any potential organic molecules. The two most fundamental requirements for life are a coded operating manual and the ability to extract energy from the environment. How are these two basic functions performed? The mechanism for storing, translating, and copying genetic information is more universal. Deoxyribonucleic acid, DNA, encodes the cell's operating manual. DNA is a polymer, a long string of nucleotide subunits bonded together. Each nucleotide comprises three parts: a sugar, a base, and a phosphate group. The deoxyribose sugar, a pentagon, links 4 carbon atoms and 1 oxygen into a ring. The base can be one of 4 kinds: adenine, guanine, thymine, and cytosine, abbreviated to A, G, T, and C, each with a skeleton of nitrogen and carbon atoms. These are the letters of the genetic code. Phosphate groups link sugar molecules, with the bases protruding to the side. If their sequence of bases matches, 2 strands aligned together bind. A always pairs with T and G with C. This forms DNA's famous double helical structure. 2 strands coiled around each other, linked by bases like the rungs of a twisted ladder. This elegant structure is the key to these molecules' information storage capabilities and reproductive abilities. The sequence of genetic letters specifies how to build proteins, and the ladder construction of the DNA means that it can act as a template for its own replication. All 3 domains of cells share the same genetic code, so it must have developed before they diverged. This is the best evidence that all life on Earth has the same root. A coordinated team of enzymes is required to copy the genetic information during cell division. Some force the 2 strands apart, unwinding the helix as they go. Others attach spare nucleotides to the exposed bases, and others join them together to form two completely new strands. A similar process creates a temporary copy of the information to form a specific protein. It unzips the DNA helix only along the right gene, base pairing and sealing RNA units instead of DNA units. RNA is virtually identical to DNA, except it has an extra oxygen atom attached to the pentagonal sugar ring, and uracil, U, replaces thymine. Thus, transcription transforms the stored DNA gene into a messenger RNA molecule, mRNA, then travels to a ribosome. RNA partially forms ribosomes, which carry out the vital cellular function of converting the mRNA code into a protein. The mRNA code comprises triplets of bases, each set of 3 specifying a single amino acid. A transfer RNA, tRNA molecule bearing the appropriate triplet attaches spare amino acids. The ribosome reads the mRNA message and attaches amino acids one after the other, recognizing them by their tRNA tag, to create the particular protein. Bonds between the different amino acids hold the protein together as it folds into its precise 3-dimensional shape. To summarize, DNA stores the information on how to make the parts of a cell, and proteins copy it every time the cell divides. DNA cannot produce proteins by itself. RNA is the middleman, conveying information from the DNA to the machinery that builds proteins. A fungus extracts energy from a tree stump by oxidizing the complex molecules that make up wood. Oxidation is a chemical process that involves adding oxygen to a molecule, or removing hydrogen and stripping an electron. Reduction, the opposite reaction, involves donating an electron to a molecule. These reactions always happen in pairs, with one molecule, the reductant or fuel, donating an electron to another, the oxidant. We often abbreviate this reduction-oxidation process as redox. All cells in the human body, including almost all eukaryotes, extract energy by breaking down a carbohydrate molecule, such as sugar glucose, and oxidizing it to produce water and carbon dioxide. Humans require oxygen to breathe, as their cells utilize this molecule as the ultimate electron acceptor. They remove electrons from the carbohydrate, transfer them through a series of intermediate molecules to carry out the cell's tasks, and finally exchange them with oxygen to generate water. This process, known as respiration, occurs in the mitochondria within human cells. The redox potential of a chemical measures its degree of oxidation. Strong oxidants, like oxygen gas, have large positive values, while strong reductants, like glucose, have very negative ones. The difference between the redox potentials of the oxidant and the reductant indicates how much energy a particular reaction will yield. An electrical battery similarly attaches the negative terminal to a reductant and the positive terminal to an oxidant. When a wire connects the two, a redox reaction begins, allowing the electron current to perform tasks. As seen in Figure 3, the complete oxidation of glucose yields a lot of energy. Cells must slowly break down the molecule step by step to release a little energy at a time. Cells convert glucose, a ring of 6 carbon atoms, into molecules of 2 carbons. This process produces energy storage molecules like ATP, whereas electron carrier molecules seize a few electrons. All eukaryotes feed the 2-carbon compound into a massive circular chain of reactions. The Krebs cycle initiates by adding the 2-carbon compound to a 4-carbon one. The cycle steadily oxidizes this molecule, releasing an electron here, producing ATP there, and snipping off a carbon here until the original 4-carbon compound regenerates, and the cycle starts again. The Krebs cycle can turn indefinitely, churning out electrons as it goes. Some of these energetic, liberated electrons are used to drive the building of complex, reduced molecules, pushing them up the hill of an organization I mentioned earlier in the chapter, but most are used to produce ATP in a very ingenious mechanism. The carriers transport the electrons to the inner wall of the mitochondria. where they deposit them at the beginning of a lengthy electron transport chain. Once on the chain, a series of redox reactions pass the electrons from one component to the next, many pushing a proton, a hydrogen atom stripped of its electrons, outside the membrane, creating a proton gradient. Each component's energy state gradually decreases until the final electron acceptor, oxygen in human cells, receives the electron. Before too long, there is a high concentration of protons outside the membrane. The cell effectively converts glucose's energy into a chemical gradient, in the same way electricity companies pump water uphill into reservoirs as an energy store. Throughout the membrane, there are specialized enzymes called ATP synthase. They allow the protons to flow back down the gradient, using their energy to drive the production of ATP, similar to the rush of dammed water spinning a turbine. The Krebs cycle uses the electron transport chain to provide 3/4 of a eukaryote's total energy demand. Researchers have calculated the overall efficiency of aerobic glucose respiration, using oxygen, to be around 40%, comparable to a coal-fired power station. Oxygen is essential for complete oxidation because it accepts electrons at the end of the transport chain, refreshes electron carriers, and stops the Krebs cycle. Some eukaryotes and many bacteria respire anaerobically, without oxygen, in fermentation, which releases about 1/10 of the stored energy. During fermentation, the Krebs cycle partially oxidizes carbohydrates, converts them into waste products, like alcohol in brewer's yeast, and expels them from the cell. If the specialty of eukaryotes is their genetic control and coordination in assembling trillion-cell constructs like the human body, the strength of prokaryotes is their metabolic diversity. Eukaryotes can, in general, only feed on things they can directly pass to the Krebs cycle, although fungi are a little less finicky, feasting on methanol, for example. But bacteria and archaea are true chemical wizards, capable of living off almost every imaginable food source. Any redox reaction, in principle, yields electrons that drive cellular metabolism. The fundamental position of electron carriers and transport chains in all cells' biochemistry implies that they probably formed the basis of the first metabolism. Aerobes are prokaryotes that use oxygen as their final electron acceptor. This powerful oxidizing agent's destructive effects poison many, referring to them as anaerobes. Some prokaryotes employ heavy metal ions, such as arsenic, as the final electron acceptor. Other cells find poisonous ions. Some bacteria even reduce uranium ions in order to run their metabolisms. Very early life may have indirectly used nuclear power to drive their biochemistry. The abundance of radioactive isotopes of uranium, thorium, and potassium would have been much higher on the young Earth than today. These unstable atoms release radiation when they decay, which can boost the surrounding molecules' energy, so they react together. Scientists believe that life in the rocks could have produced and metabolized a significant amount of reduced organic molecules in this way. Such cells would have effectively fed off nuclear reactions. Thus, many prokaryotes can derive their metabolic energy from inorganic reactions. They don't need to break down complex organic molecules like glucose. Some don't even need access to organic molecules as a carbon source for their cellular building blocks, amino acids, sugars, bases, and so on. They are self-sufficient and can build everything they need from scratch, turning basic compounds like carbon dioxide into all the biological molecules they require. For example, a human cell, which needs everything handed to it on a plate, is an organoheterotroph. A bacterium that survives in organic ion reactions but needs an external supply of sugars is a lithoheterotroph. In contrast, a chemical autotroph uses inorganic substances for energy and carbon. Carbon fixation converts inorganic carbon dioxide into organic molecules and moves them up the energy gradient. Some chemoautotrophs live underground, extracting the ions and carbon dioxide they need from the surrounding minerals and eating rocks for breakfast. We'll look at some of these amazing organisms in the next chapter, discussing extremophiles. Many potential niches for life in the solar system are far from the sun and devoid of pools of ready-made organic compounds, so chemoautotrophs are the odds-on favorites for extraterrestrial life. The table above shows a third energy source. Photoautotrophs use sunlight's energy to run their metabolism and fix carbon dioxide into carbohydrates. This photosynthesis process is enormously important in Earth's biosphere. Like respiration, the key to photosynthesis is the liberation of electrons to perform work for the cell. Photosynthesis has two essential parts. The first captures light energy, and the second uses that energy to drive carbon fixation. Chlorophylls, a class of magnesium-containing compounds, are central to the first part. These molecules are extremely efficient at absorbing visible light, using their energy to excite electrons within their structure. Carriers pick up these electrons and shuttle them to an electron transport chain. Modern plants have two photosystems for trapping light. The first photosystem is a cyclic pathway that transports the released electrons down a transport chain, then returns them to the chlorophyll. This is an ancient mechanism. A similar system provides energy for many photosynthetic bacteria. In the second photosystem, light energy excites electrons, sending them down a transport chain or storing them in a carrier to drive later reduction reactions. But it also causes the splitting of a water molecule. This splitting releases a low-energy electron to replace the one the photosystem lost. The photosystem releases the other byproduct, oxygen, as waste. Therefore, we call this process oxygenic because it generates oxygen gas. Like respiration, the electron transport chain in both photosystems generates a proton gradient for ATP generation. The second part of photosynthesis, using the captured energy to build organic molecules, does not require light. In a crucial carbon fixation step, the Rubisco enzyme attaches a carbon atom from carbon dioxide to a 5-carbon sugar. This 6-carbon sugar then breaks in half to give two 3-carbon molecules that enter the start of a powerful reaction loop, effectively reversing the Krebs cycle. ATP and the carriers generated from the photosystems pump energy and electrons into the cycle, driving the reactions. The plant siphons off some products as activated 6-carbon sugars, creating all the complex carbohydrates it needs. Ultimately, the plant recreates the 5-carbon sugar, restarting the cycle. Photosynthesis generates ATP and fixes carbon dioxide into carbohydrates, gaining the electrons it needs by splitting water to release oxygen. Thus, both the fundamental demands of the cell, biochemical energy and reduced carbon compounds, are simultaneously satisfied. The evolution of photosynthesis and its effect on the atmosphere were critical to the history of life on Earth. Today, Earth's photosynthetic organisms fix about 10,000 billion metric tons of carbon annually, supporting the entire surface biosphere. One of the planet's most abundant enzymes, Rubisco, leaves a distinctive signature in the carbon it fixes, a biosignature that ancient rocks can use to date the emergence of oxygenic photosynthesis. The Earth's crust has a natural negative redox potential. The atmosphere and oceans are slightly more oxidized by sunlight and radiation. This creates an electrochemical gradient or potential difference between the crust and the water and air above it, a planet-sized battery. Many lithotrophs extract energy from this, living right on the interface, reacting the reduced ions in the crust with oxidized ones from above. As highly reduced organic substances accumulate as sediment over the land and ocean floor, photosynthesis steepens this energy gradient and pumps oxygen into the air and water. Over evolutionary time, the potential difference for the entire planet has increased. Many redox reactions occur naturally, so the only way life can use them to extract energy is to run them more quickly than it would normally proceed. Once the chemicals achieve equilibrium, the energy gradient vanishes, leading to the extinguishment of life. Enzymes are crucial. They allow life to outrun the geological processes that would eventually remove the gradient. For example, the rusting reaction of ferrous iron oxidation typically takes years, so enzymes can easily improve and keep up. Some reactions only occur at low temperatures with the action of life, while others, such as the one between iron oxide and hydrogen sulfide, proceed so quickly that enzymes cannot perform any better. This need to accelerate abiotic reactions leaves a distinctive signature in the environment. If you descended deep below the surface of the Black Sea and looked at the concentrations of different chemicals as you went, you would see a series of sharp gradients and peaks. Oxygen is present at high concentrations near the surface, but rapidly disappears due to aerobic respiration. Lower down are peaks of reduced nitrogen and iron caused by chemoautotrophs, and deeper still are rising levels of other reduced chemicals, such as ammonia and hydrogen sulfide. Life is necessary for the reactions producing these chemicals to run too slowly to set up gradients like this. Diffusion and water currents would mix them more quickly than they could form. The fact that the Black Sea is not an even mix of chemicals is an ironclad proof of biological processes. All ecosystems show such tiers at different scales, across meters in lake water, centimeters in sediments, and micrometers in biofilms, such as on the surface of stromatolites. This allows astrobiologists to detect alien ecosystems without making any assumptions about the appearance, genetics, or metabolic networks of the organisms within them. Life betrays its presence in a layered series of redox zones in the environment, indicating the driving force of biologically accelerated chemistry. Is there another way? What makes us believe that life in another world will be like life in this one? First, many of the building blocks used in terrestrial life, such as sugars, amino acids, and bases, are extraterrestrial. The massive dust clouds between the stars and during the solar system's formation produce these compounds. Could we combine these subunits, or those slightly different from the ones chosen by terrestrial life, in different ways to form living systems? Since there is enormous metabolic diversity within terrestrial life, but only a single genetic system, we'll look at RNA. Is the particular makeup of terrestrial RNA and the closely related DNA unique, or could a similar molecule do an adequate job? Finding carbon and water-based life is the assumption that drives our current exploration of the solar system, but how solid is the reasoning? Let's first examine RNA's molecular cousins more closely. Prebiotic chemistry would have produced various sugars and bases during the first stage of life's emergence, long before cells, when only replicating molecules inhabited the Earth. So why does all current life on Earth use only two closely related polymers to store its genetic information? What's so special about the particular subunits used in RNA and DNA? Are they superior to the alternatives, or was their survival more of a fluke, an evolutionary happenstance? The RNA system's random selection may have led to its prevalence today, making it difficult for evolution to reverse. This resembles how mechanical typewriters retain the QWERTY key layout in computer keyboards. This pattern's design could be more optimal, which forces slower typing and reduces the number of jams. After numerous attempts to change the value, we have fixed this suboptimal system, which proved too difficult. Researchers have been creating artificial alternatives to address whether there are workable substitutes for RNA and DNA. DNA and RNA are essentially the same, with the only differences being an extra oxygen bonded to the ribose sugar ring, the use of the T base instead of the A sugar, and a phosphate group that binds the sugars into a strand. Changing the bases within a DNA strand is relatively easy. Experiments have systematically substituted lab-made bases by changing the letters of the genetic alphabet. The structure of the DNA is not overly distorted. It still forms a double helix despite the bonding between the base rungs being different. Prebiotic processes believed to occur in interstellar gas clouds or on primordial planets are unlikely to produce these non-standard alternatives in alien DNA. Most experiments into alternatives focus on RNA, as it is thought to predate DNA in life evolution. Scientists have tested two main modifications to terrestrial RNA: attaching the bonds linking neighboring sugars to different carbon atoms within the ring, or using a different sugar molecule. Phosphate groups bind the third and fifth carbon atoms of adjacent sugars to form our RNA into a long strand. An alternative would be to link the second and fifth carbons. However, not only does the formed strand become much more vulnerable to snapping, but it also severely weakens the strength of pairing between bases. Different isomers of ribose exist, each made of the same atoms, but with a slightly different configuration. Researchers have found that RNA derived from other isomers exhibits exceptionally strong base pair bonds, resulting in more stable helices than native RNA. Another fascinating fact is that a group of different isomer alternatives help strengthen RNA structures over time. This implies that natural RNA was chosen for more than stability. A moderate base pairing strength is better because it allows easier separation of the two strands during replication. Furthermore, helices with stronger base pairing tend to make more mistakes when binding sequences that don't match perfectly. RNA can undergo a final major modification by changing its sugar type.
Ribose is a 5-carbon sugar. What about swapping it for one with 4 or 8 carbons?
Upon completing the experiments, researchers discovered that 6-carbon sugars, like glucose, do not function well as information storage polymers. The bonding between the base pairs was woefully inadequate. Not only was it very weak, but the bases were much less particular about whom they paired with. A would happily bond with G, rather than only with T. The larger sugar units are probably too bulky and interfere with close bonding. The results from 4-carbon sugars were much more exciting. The polymer TNA, made from sugar 3-Os, allows base pairing with the same stability and accuracy as RNA, and forms a perfect double helix. TNA can even cross-pair with RNA and DNA, which was impossible for most alternatives. This has raised a lot of interest due to the simpler nature of TNA, which is based on 4-carbon sugar. Such a sugar would form more easily in prebiotic reactions, so TNA is much more likely to have arisen on the early Earth. This will become clear when we examine the evolution of replicating molecules and the difficulties in explaining how DNA or RNA originated in Chapter 4. So far, experiments into worthy competitors to RNA have led to many dead ends. Many alternative polymers cannot form stable double helices or are inferior to RNA in other ways. At the moment, alien life evolved from a soup of prebiotic chemicals similar to those on early Earth and is likely to use either RNA or a very similar molecule to store its genetic information. However, we have yet to explore the vast landscape of potential alternatives to the sugar, bond placement, and base of RNA-DNA, let alone a completely different kind of polymer. The next step is to test which polymers can replicate and develop over time under Darwinian evolution. Polymeric chemistry is essential for our lives. It stores and transmits information using nucleic acid polymers, DNA, and RNA. We use carbohydrates, such as starches, as polymers to store energy. The amino acid polymers and proteins are the most diverse. Carbon forms the backbone of all these molecules. Life on Earth would be impossible without it. Given its inherent nature, building molecules with the necessary complexity for life is challenging. Silicon is the most frequently mentioned alternative. On the periodic table of elements, silicon sits directly below carbon, exhibiting similar behavior to carbon in chemical reactions, such as the ability to form 4 bonds simultaneously. Because the silicon atom is larger than the carbon atom, it forms less in the star's cores, resulting in a greater abundance of carbon in the universe than silicon. However, due to the process that created the solar system, silicon is much more abundant on Earth, almost 30% of the crust, so the element silicon is unlikely ever to be in short supply on a rocky planet. Considering this, if complex silicon chemistry were possible on Earth,
it ought to have resulted in life based on silicon rather than its rarer chemical cousin, carbon. Terrestrial life uses silicon, such as in the protective shells built by diatomic algae, but it does not form the basis of our meric or metabolic chemistry. In another sense, silicon, a larger atom, is important. Its bonds with other atoms are generally weaker than carbon, producing very fragile polymers, at least under the surface conditions prevalent throughout Earth's history. Under very different conditions, such as high pressures and high or low temperatures, silicon may indeed form sturdy enough polymers. However, we need to find out what such a biochemistry would be like, making designing tests to look for it impossible. Another issue with silicon is the compound it forms with oxygen. Carbon polymers undergo oxidation during carbohydrate respiration, releasing carbon dioxide gas. The analogous process would produce silicon dioxide, which is sand. This substance is hard, insoluble, and solid, making it challenging for life to thrive. The final reason to favor carbon-based life, at least in our corner of the galaxy, is that organic building blocks are rife throughout the heavens. Falling vital carbon compounds are believed to have smothered primordial planets early in their development. Since space does not exhibit similar silicon compounds, searching for carbon-based life is the only logical action. We need to clarify whether extraterrestrial life would depend on liquid water. Water has an impressive list of skills. First, liquid water is an exceptionally effective solvent. The two hydrogen ends of its V shape are positively charged, whereas the oxygen atom at the point is slightly negative. The water molecule is polar. It has both positively and negatively charged ends. This means that water molecules can form weak hydrogen bonds with each other and other polar substances or ions in order to dissolve them. Having molecules available in a dissolved state is crucial for the rapid chemical reactions of life. It also helps gather essential nutrients and expel waste products. Molecules caged up in a solid state prevent organic life from moving and reacting, while a gaseous life form faces the challenge of rapidly moving away from each other and never reaching concentrations high enough for swift reactions. The hydrogen bonds in water ensure that it is liquid and available for biology across a wide range of temperatures. Water is also peculiar in that it expands when it freezes. Ice is less dense than liquid water, so it floats on top of it and insulates it, preventing the entire volume from freezing solid. Water can also absorb much heat energy without raising its temperature, which could damage biological molecules. This makes it an excellent thermal buffer that protects cells from wild temperature swings and prevents disrupting vital activities like enzyme action. Water is also chemically useful, contributing oxygen or hydrogen atoms across various biochemistry processes, such as the hydration reactions that break down proteins or starch polymers into their subunits. No other known liquid combines all these amazing properties, but we need to know which ones are relevant to the emergence of life. Could a different solvent, even one with fewer attributes, still be suitable for biochemistry? For example, people assert that water polarity is critical for protein folding and operation. Peptoids, conversely, can fold up in pure methanol, which is chemically very similar to proteins. We may even be asking the question the wrong way around. The properties of water seem fortuitously fine-tuned for life, but it is equally possible that life on Earth is fine-tuned for a watery environment. Because water is our planet's only solvent available in large quantities, terrestrial life evolved to exploit its specific properties. Terrestrial life is better suited to water than vice versa, and there may be no beneficial chemical reason why life can't emerge in other solvents. In fact, water is inappropriate for life. It is fairly reactive and tends to break apart complex organic molecules. RNA, for example, is particularly short-lived in water. What alternatives are there? Ammonia is a common compound in the galaxy. It is present in outer space, dust clouds, and liquid droplets in Jupiter's clouds. A mixture of water and ammonia can exist as liquid at temperatures much lower than pure water, forming a hybrid solvent. This is especially important because there may be water and ammonia aquifers beneath the surface of Saturn's moon, Titan. Another chemical, formamide, is liquid across various temperatures and pressures, dissolves salts, and has other properties similar to water. Other possible solvents include methane and liquid nitrogen. However, although they could support organic biochemistry, the much lower temperatures are a big issue. In such cold climates, even if the solvent is liquid, the chemistry will differ. The colder the conditions, the slower the processes occur, and the more effective an enzyme must be in getting reactions to proceed at a useful rate. At these temperatures, biochemical reactions may proceed so slowly that they render life impossible. A non-water-based ecology is so alien, like non-carbon-based life, that designing experiments or instruments to send to other worlds is difficult. We have yet to begin to engineer an alternative metabolic network within water, let alone one based on an entirely different solvent with chemical properties we've barely researched. NASA's astrobiological motto is "Follow the water," because there's life on Earth where there's liquid water. This is likely to remain the focus of astrobiology research for now. Hunting in conditions we know can support life rather than speculating about other, perhaps fantastical, possibilities. What do we know about life? We know it must exist on an energy gradient, extracting energy from the environment to push its complexity uphill. Two fundamental energy extraction methods power life on Earth. Electrons liberated from photosynthesis, or redox reactions, generate a proton gradient that drives ATP synthase or ferments reduced organics, directly producing limited amounts of ATP. Life must also possess a coded self-description that is replicable for future generations. Terrestrial life fulfills these requirements through polymeric chemistry, necessitating the dissolution of organic substances like carbon in a fluid medium like water. While other solvents are feasible, the versatility of carbon is believed to make it a universal component of chemical life. The precise polymers that extraterrestrial life could construct using carbon may become more apparent. Cosmic requirements. 13 billion years ago, the youthful universe was completely dark. The searing light from the fire of its conception, the Big Bang, had long since dissipated as the universe expanded. The initial burst of pure energy condensed to form subatomic particles, protons, electrons, and neutrons, which assembled themselves into the simplest elements within the first few minutes. The Big Bang mostly produced hydrogen, some helium, and minuscule amounts of lithium. The splendid periodic table of elements contains the 91 naturally occurring elements we have discovered on Earth, with only the first 3 boxes filled in. Life depends on heavier elements for constructing complex polymers such as DNA or proteins, and building the rocky planets or moons that provide a home. The early universe was not only completely sterile, it needed to be more complex, in atomic terms, to provide the basic chemistry kit of life. The Big Bang left its imprint on mass distribution as thin wisps of matter threaded through vast voids of dark emptiness. In time, these gossamer veils collapsed under the force of gravity into long strings of galaxies. Massive clouds of gas were collapsing within these galaxies, becoming hotter and hotter until nuclear fusion reactions flickered into action, flooding the universe with the first light it had seen since its creation. The universe's first stars were the candles on its billionth birthday cake. The immense heat and pressure in the cores of these first stars forced atomic nuclei together. This process, nuclear fusion, unleashes enormous amounts of energy and is what keeps stars burning. Even more importantly, at least for the future development of life, fusion creates heavy elements from the primordial matter created in the Big Bang. The nuclear furnaces in the hearts of the first stars formed the atoms crucial for life. They are the original alchemists, converting hydrogen into the full diversity of the periodic table. First, the center consumes hydrogen nuclei and single protons, squashing them together to produce helium nuclei, which have 2 protons. This stage of nuclear fusion proceeds until the center begins to run out of hydrogen fuel. The star finds itself with a core of helium ash, surrounded by a layer of hydrogen that continues to burn. The outward pressure from the internal fusion no longer balances the enormous weight of the star above, leading to further compression of the core, resulting in even higher temperatures and pressures. As the core shrinks, the outer envelope of the star's atmosphere expands, producing an enormous bloated star that is hundreds of times larger than our Sun and relatively cool on the surface. These aged, swollen stars, red giants, have a core temperature of around 100 million degrees. Helium begins to undergo fusion at this temperature. A triple alpha process reaction occurs when 3 helium nuclei, known as alpha particles, fuse together to create carbon. Another helium is added, converting carbon into oxygen. The triple alpha process deserves a longer explanation. The reaction's energy greatly enhances the addition of the 3rd helium nucleus, nearly matching the energy of an excited carbon state. This phenomenon, resonance, significantly accelerates carbon production through nuclear fusion. This correspondence in energy levels is a cosmic fluke. If some of the physics constants were even minutely different, stars would not forge carbon. The universe has been tailor-made in some ways, with certain factors finely tuned to allow for carbon production and life. This does not imply the existence of any purposeful design. It is merely the consequence of observational bias. Had the universe been created with slightly different rules that perhaps did not allow carbon synthesis, our life would have been impossible, and we would not have witnessed such a different universe. The exquisite arrangement of things to promote life, known as the anthropic principle, should not surprise us. When the available helium runs out, fusion in the star's core grinds to a halt once more. The star's interior now resembles a hot onion, with a core of carbon and oxygen encircled by a helium shell and an outer layer of hydrogen. Red giant stars compress their cores further, undergoing another round of fusion that yields elements like silicon, sulfur, and iron, a large nucleus with 26 protons. Iron is the end of the road for nuclear reactions in the star core, as its nucleus is too stable to release any more energy when it undergoes fusion. The star ultimately perishes due to this iron heart. The outer layer's massive weight crushes the core, and the renewed fusion's outward pressure cannot counteract this compression. As the constituent parts of the nuclei, the electrons and protons, force together, the stellar collapse continues until the internal pressure reaches extreme levels. The core can compress no further, and the unimaginable heat generated causes the outer layers to detonate violently outwards, ripping the star apart in the colossal explosion of a supernova. This single star briefly outshines the rest of the galaxy during its death throes. The event is so energetic that a final round of fusion reactions occurs, involving even the iron nuclei forming the universe's heaviest elements. The star launches its outer layers into space, dispersing the biologically essential heavier elements throughout the universe. Oxygen soon combines with the hydrogen of interstellar clouds in the ratio of 1 to 2. This is the chemical formula of water, which is quite literally the liquid of life, and so these first stars gradually turned the galaxy wet. Terrestrial biology uses some other new elements, such as copper and iodine, but the significance of the very heavy elements lies in the radioactive instability of many of their isotopes. Elements like uranium are not directly relevant to building biological molecules, but play a crucial role in developing rocky planets. Radioactive decay helps drive volcanism and plate tectonics on Earth, and it may even have created some organic molecules that the emerging life relied on. The first stars were massive, hundreds of times faster than our Sun. Heavyweights burn through their nuclear fuel at prodigious rates, quickly exhausting their resources and reaching an explosive endgame. The first stars typically lasted only a few million years, less time than our species has taken to diverge from the chimpanzees. Their deaths blasted out material that floated through interstellar space, collecting together as loose clouds of gas, dust, and nebulae. These clouds, loaded with heavier elements, collapsed under gravity to form new stars. This process went through several rounds, with each generation of stars processing the gas through nuclear fires. Our galaxy gradually became richer and richer in the elements capable of intriguing chemistry. Therefore, the heavier elements greatly boost the most recent stars. Astrophysicists refer to any element heavier than helium as a metal, much to the dismay of chemists or blacksmiths. Our local star, the Sun, is a member of this latest breed. Our solar system formed from a nebula loaded with heavy elements produced by the Sun's ancient ancestors. While it is true that all life originates from stardust, it also forms the foundation of Earth's rocks, oceans, and air. In fact, a small proportion of the hydrogen atoms that make up the molecules of your body don't come from this galaxy. The sheer violence of supernova detonations ensures that some material is ejected right out of its home galaxy. The Milky Way has picked up hydrogen from the outer envelopes of exploding stars in our next-door galaxy, Andromeda, and incorporated it into nebulae here. Without wanting to overstrain the sentiment, we are not only children of the stars, but also partially intergalactic beings. The first generations of stars in the universe played a critical role in forging the elements needed by life. The six most important are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, sometimes abbreviated to CNOPS. Others, like sodium and several other metals, are also crucial in small amounts. But the rudimentary building blocks of cells are organic molecules, the amino acids that make up proteins, the nucleobases of DNA or RNA, and simple sugars. Even these organic molecules could be ubiquitous in the galaxy. Star-forming nebulae are enormous and typically contain enough mass to create millions of stars the size of our Sun. Such clouds are 99% gas, but contain small solids, microscopic dust particles, and ice crystals. The gas is mostly hydrogen, but thanks to the fusion furnaces of previous generations of stars, other atoms, such as carbon, oxygen, and nitrogen, are also present. Over the millennia, these lone atoms have bonded together to create simple compounds such as water, carbon monoxide, ammonia, and hydroxyl. The first stars to form in the nebula light it up from within. The Orion Nebula, the middle point of the constellation's sword, glows a vivid red from the hydrogen it contains. Simple molecules and galactic radiation absorb the light, particularly UV, emitted by these hot young stars, increasing their reactivity. Much of the intriguing cosmic chemistry occurs in the denser regions around new stars, where the temperature approaches about 170 degrees. The solid particles, although a tiny proportion of the total mass of the molecular clouds, play a large role in astrochemistry. The solid surfaces can absorb reactive chemical species, acting as catalysts to bring different molecules together for reaction. The molecules recycle endlessly between the gas and solid phases, evaporating back into the free-floating nebula and reabsorbing onto an icy dust particle. Dust particles play an important role in shielding the deep interior of the nebula from too much heat or UV radiation from the maturing stars. UV light is a double-edged sword. It provides the energy to allow simple molecules to bond, but is destructive of more complex molecules. Over time, more complex organic molecules build up within the dust clouds, although they are always 1,000 times less abundant than the hydrogen. Scientists have identified approximately 130 distinct molecules, with the most intricate consisting of up to 13 interconnected atoms. Many of these, such as ammonia, formaldehyde, and hydrogen cyanide, are considered crucial in the prebiotic chemistry in Earth's seas. These interstellar clouds' most common organic molecules are polycyclic aromatic hydrocarbons, PAH, with a carbon atom backbone arranged in various configurations. One example is naphthalene, the chemical that gives mothballs their distinctive smell. They are compounds that escape in soot particles from car exhausts and are unimportant to terrestrial life. However, it is very simple to change PAH chemically into the porphyrins and quinones that comprise the metal-holding centers of many biological molecules, such as metabolic enzymes, the active centers in chlorophyll and hemoglobin, and parts of the electron transport chain. The formation of these precursors is central to the development of life on Earth. Scientists have not unambiguously detected anything as complex as the simplest amino acid, despite discovering numerous simple organic compounds in these vast gas and dust clouds. Finding these complex compounds is challenging due to their relative rarity and the need to be deep within the cloud for protection from UV radiation. Lab experiments have produced glycine, alanine, and serine, the three most abundant amino acids in earthly proteins, by replicating the cold gas, dust particles, and UV levels of nebulae. However, the most compelling evidence for the cosmic origin of more complex organics comes from a specific class of space rocks. Carbonaceous chondrites are a type of rock from the asteroid belt. Scientists believe they contain a small percentage of carbon compounds, remnants of the primordial material that formed the planets. One example is the Murchison meteorite that landed in Australia in 1969 and underwent extensive analysis. It has an amazing variety of organic molecules, including different sugars, the 5 nucleic acid bases that DNA and RNA use on Earth, and chains of fatty acids that, when mixed with water in the right conditions, form a hollow crystal about the size of bacteria. However, the most surprising finding is that more than 70 amino acids exist. Only 6 of the 20 different amino acids used by terrestrial proteins in the Murchison meteorite indicate that the vast majority are completely alien to life on Earth. This last finding is not the only convincing proof that the organic substances present aren't simply contamination from terrestrial life since the meteorite landed. The sugars and the amino acids in the rock are both enantiomers, which is a sign of abiotic chemical reactions. On Earth, life enzymes produce only one enantiomer of an organic molecule. However, there is a slight, intriguing bias towards the left-handed enantiomer in the Murchison amino acids. The origin of this bias is unknown, but it is intriguing why terrestrial life favors one antagonist over the other. One theory suggests that the polarization of UV light from nearby stars affected the complex amino acids formed within the interstellar clouds before the solar system's birth. If true, the enantiomer bias displayed by cells on Earth may be an imprint of this ancient starlight. The galaxy seems to be a very encouraging place for astrobiology. The building blocks of terrestrial biochemistry are apparently widespread throughout the cosmos. How all these raw ingredients arrived on Earth is a topic I shall cover in the next chapter. We'll examine the type of planet required to transform this prebiotic soup into living matter. Liquid water is considered absolutely necessary for life, as I discussed. Prokaryotes have colonized practically every moist niche on Earth, and while water alone is not sufficient for life to evolve, it is certainly considered a crucial prerequisite. For a world to evolve life, it must, first and foremost, have expanses of liquid water. It was believed that a large rocky planet with a hot core, thick atmosphere, and surface oceans, a terrestrial planet, was necessary. Later, I will discuss the possibility of life on worlds other than warm, rocky planets. But for now, I will look in detail at what Earth-like life probably requires. The planet's surface temperature and pressure determine whether groundwater is liquid. These axes allow us to plot the conditions under which water flows on a graph, as shown in Figure 5. If we gaze halfway up at 1 atmospheric pressure, the sea level pressure on Earth, we can trace our eyes horizontally across increasing temperatures. We see the transition from solid to liquid, melting at the familiar 0 degrees C and boiling at 100 degrees C. Increase the pressure, such as at the bottom of the ocean, where water is still liquid at over 300 degrees C. On the other hand, If you decrease the pressure below the triple point, the temperature and pressure at which all three phases, gas, liquid, and solid of water may coexist, then water cannot exist in a liquid state at all. It sublimates straight from solid to gas. If, for the moment, we assume that an alien world has an atmosphere as thick as our own, we can see what range of temperatures will support liquid water. The world's orbital proximity to its star largely determines the surface temperature. If the water is too close to the central heat, it boils off. If it is too far away, it becomes permanently locked up as ice, rendering it incapable of performing biological functions. The range of distances that allows liquid water is known as the stellar habitable zone, HZ. This is like a thin ring circling the star within which the planet must orbit. People sometimes refer to this as the Goldilocks principle, stating that life requires a planet with just the right temperature, neither hot nor cold. However, this is a very simple look at what determines the presence of liquid water in a world. The planet's atmosphere plays an enormous role in regulating the surface temperature and pressure. We'll now turn our attention to this. Earth's thick atmosphere insulates the surface very effectively. Carbon dioxide, methane, and water vapor in the air act as greenhouse gases, allowing the visible part of sunlight to heat the Earth's surface so that it begins to give off infrared or thermal radiation. However, such gases absorb these lower wavelengths of light, trapping the warmth below, an effect identical to the panes of glass trapping heat within a garden greenhouse. The greenhouse effect is a perfectly healthy part of the planetary system. If it weren't for the greenhouse effect, life on Earth's surface would be impossible, as the average temperature would be more than 30 degrees lower and the surface a frozen wasteland. The concern is that humans are now pumping so much carbon dioxide into the atmosphere that it might begin tipping the climate away from its current stable point. As I briefly mentioned in the introduction, carbon dioxide in the atmosphere is not static, but one stage in a massive loop of carbon moving through the planet's rocks, water, and air, which is the carbonate-silicate cycle. Dissolved in water, carbon dioxide forms a mild acid, carbonic acid, which weathers silicate rocks to form the mineral calcium carbonate or chalk. The ocean floor deposits this rock, thereby securing the carbon. A process that returns carbon to the air in a few 100 million years would remove all carbon dioxide from the atmosphere. This would be catastrophic for the climate, as an important greenhouse gas steadily disappears and the world freezes. Plate tectonics provides the return leg of the cycle. On Earth, new crust produced at the ocean ridges migrates slowly outwards, accumulating thick layers of carbon-containing sediment. Subduction zones push the crust inexorably downward, causing it to melt in the hot interior mantle. Volcanoes release carbon dioxide once more, venting it into the atmosphere. Most crust completes the journey in about 150 million years, steadily recycling carbon dioxide into the air. Most importantly, this particular elemental cycle is self-regulating. The rate at which silicate rocks weather to form chalk varies depending on the temperature. Warmer conditions accelerate carbon dioxide removal from the atmosphere, diminishing the greenhouse effect. In a colder climate, the reaction slows and carbon dioxide accumulates, raising the temperature. This negative feedback automatically keeps carbon dioxide levels in check, as though the entire planet has a thermostat that carefully regulates the global climate. The oceans are vital in providing an environment for life, but the water also performs a crucial role in controlling the temperature, eroding silicate rocks, and lubricating the motions of the tectonic plates overhead. The carbonate-silicate cycle regulates the complex interplay between the star's brightness and the insulating properties of the planet's atmosphere, which determines the stellar habitable zone. Seas of liquid water and carbon dioxide in the air are critical to the functioning of this climate control system. These two factors really define the inner and outer boundaries of the ecosystem. The process that acts to lower the surface temperature can only do so much. If the planet is too warm, large amounts of water vapor, a greenhouse gas, evaporate into the air. But unlike carbon dioxide, water vapor has no negative feedback condition, so the world gets hotter and hotter. Eventually, the atmosphere becomes laden with steam, and the oceans boil dry. Without the lubricating effects of the water, the conveyor belt of plate tectonics creaks to a halt, and the carbonate rocks exposed on the surface decompose in the heat to release their carbon dioxide. UV light from the star splits apart, photon dissociates. The water vapor is high in the atmosphere, and the lighter hydrogen gas escapes the planet's gravity from deep. The planet permanently loses its water inventory, thereby forfeiting its potential for life development. The HZ's inside limit is the distance from a star that triggers this catastrophic runaway greenhouse process. On the other hand, if a planet is slightly too far away from its star, even a thick atmosphere cannot provide sufficient warming. The oceans begin to freeze, creating wide areas of pure white on the planet's surface that reflect more of the sun's warmth. The global temperature nosedives. Eventually, even carbon dioxide begins to freeze out of the atmosphere. As this crucial greenhouse gas begins to deposit as frost on the ground, all hope for climate recovery disappears. Such a fate, a runaway glaciation, sets the HZ's outside limit. For a planet to remain pleasant long enough for life to emerge, it must skirt gingerly between the twin dangers of runaway greenhouse and glaciation. Earth has obviously managed this for the last 4 billion years. We have built computer simulations of Earth's climate to calculate the extent of the Sun's HZ. The average distance of the Earth's orbit from the Sun is known as the astronomical unit, AU. Estimates for the current HZ range from a generous 0.7 to 1.2 AU to a very narrow 0.958 to 1.004 AU, depending on the assumptions made about various factors. Either way, the habitable zone around a star is very thin, like a knife edge on which Earth is precariously teetering. The type of orbit the planet follows is also important. Earth's orbit around the Sun is almost circular, but could life survive on a planet with a much more elliptical path that swoops close to its star before swinging further away? Despite the possibility of the planet being too close to other planets' gravitational pull, potentially forcing it out of the Hermit zone, such an eccentric orbit presents significant climatic challenges. The meandering world would edge towards a runaway greenhouse effect on one part of its path and runaway glaciation on the other. Even if the entire orbit is still within the bounds of the HZ, the wild swings in heat input might make for an erratic climate, preventing the stable conditions necessary for life. However, large oceans might act as an effective temperature buffer, slowly warming up and storing summer's excess heat for release during the outer region of the orbit. Developing a solar system with a rocky planet within the star's HZ is only part of the problem. The bounds of the HZ are not static, but move as the star matures and grows steadily hotter. Over time, the ethereal ring of safety around the star sweeps outwards. For example, over Earth's history, the Sun has brightened by about 25%, but the average global temperature has remained remarkably constant, thanks to negative feedback loops like the carbonate-silicate cycle. However, this negative feedback can only compensate for so much. At some point in Earth's future, the aging Sun will brighten to such a degree that the inner edge of the HZ will sweep right past our planet. The atmospheric thermostat breakdown will initiate the inexorable process of a runaway greenhouse. This fate won't befall the Earth for another billion years or more, but the rate of solar brightening depends on the size of the star. The more massive a star is, the more quickly it burns, and therefore, the less time a planet has before it slides out of the habitable zone. The continuously habitable zone requires a planet to have the right orbit and a suitable, long-lived parental star. Terrestrial planets orbiting stars 20% or more massive than the Sun are unlikely to have enough time to evolve life. stars smaller than the Sun smolder much more slowly, providing ample time for life to develop. However, scientists are still determining whether such cooler stars are feasible astrobiological targets due to their numerous problems. Size matters when it comes to a planet's habitability, as does a stable orbit within its star's continuously habitable zone. In certain aspects, the Earth stands out among the planets of the Sun's family. It is the largest rocky body in the solar system, and the only one with a detectable planet-spanning ecosystem. Since, so far, we have detailed information about only this one solar system, we cannot tell whether this is merely a coincidence or if the Earth is abnormally large enough to have permitted the development of life. Astrobiologists have excellent reasons to think life might require a roughly Earth-sized planet. A planet much smaller than Earth would have too weak a gravitational grip to cling to a thick atmosphere for the time required to evolve life. A thin atmosphere would provide only limited greenhouse heating, and the lower air pressure would limit the temperature range of flowing water. Thus, a small world would face difficulties sustaining liquid conditions long enough for life to evolve. Small planets also lose their internal heat much more quickly. The Earth's core is still scorching from its formation, and the mantle above is fluid enough to allow the convection currents that drive plate tectonics. This geothermal energy ensures that the Earth continues to be very volcanically active. Plate tectonics and volcanism are critical for the carbonate-silicate cycle that stabilizes global temperature. Another important function of the Earth's hot interior is that the churning iron within its core induces a powerful magnetic field. This force field reaches high above the top of the atmosphere and safely deflects the solar wind, a fast flow of particles streaming off the Sun's surface. On planets without this protective cocoon, the solar wind slowly but steadily blows away the atmosphere. Mars is believed to have lost a large volume of its original gases and water into space, partly because its magnetic field failed almost instantly after the planet's formation. Thus, for several reasons, planets with a thinner atmosphere and cooler interior would be much more vulnerable to the threat of a runaway glaciation. Small planets would not have been hot long enough to create a differentiated interior. The inside of the Earth is layered. The heaviest elements, such as iron and nickel, have sunk to the core, which is covered by a mantle of molten silicate rocks under a thin, solid crust. The decay of radioactive isotopes such as uranium, thorium, and potassium, as well as heavy, unstable nuclei produced in previous supernovae, releases heat that drives tectonic shifts and volcanism. These radioactive elements must concentrate in the planet's core for long periods of geothermal activity. Otherwise, their heat would dissipate rapidly. A dwarf Earth would face several challenges in developing life. Its lower gravity and thicker, more rigid crust would allow for higher mountains. However, the loss of atmosphere and water would lead to cold surface conditions and shallow seas. For such a world, runaway glaciation is a likely fate. A giant Earth would encounter two main problems. First, the hotter interior would mean high volcanic activity, with a continual spew of greenhouse gases out into the atmosphere, causing higher surface temperatures. A large planet's greater gravitational pull would also retain a much thicker atmosphere, contributing to higher temperatures. Second, the planet could run into serious problems with its own geography. A hotter interior means a thinner, more pliant crust, which, combined with the stronger gravity, would make the surface relatively smooth with little relief. Little of the atmosphere or water would have evaporated due to the higher gravity and probable strong magnetic field. These conditions favor a deep, planet-wide ocean with little dry land. The lack of exposed rock would mean a very low silica erosion rate. Without an effective mechanism to remove excess carbon dioxide from the atmosphere, the carbonate-silicate cycle would break, causing its levels to rise inexorably. Thus, a large terrestrial world would provide the perfect conditions for a runaway greenhouse effect, ultimately boiling off the oceans and sterilizing the face of the planet and any life that may have emerged. Therefore, the world's mass significantly influences the stellar HZ and the details of the planet's atmosphere. Our two neighboring planets, Venus and Mars, perfectly illustrate this point. Venus has a heavy atmospheric blanket of carbon dioxide and a hellishly hot, bone-dry surface. It's a clear example of a world with runaway greenhouses. Mars is bitterly cold, with a paper-thin atmosphere and an effortless diagnosis of runaway glaciation. Intriguingly, if the positions of the two planets had been swapped at the solar system's birth, with the smaller Mars closer to the Sun and Venus further away than Earth, their development may have turned out very differently. Despite its thinning atmosphere and reduced volcanism, Mars may have stayed warm enough to retain seas on its surface, and Venus may never have tipped over the threshold that sent its greenhouse spiraling out of control. Instead of just Earth, the solar system might have had a family of 3 habitable planets. The Earth has one other quirk important for survival, particularly multicellular animal life, the largest moon of all inner planets. Neither Mercury nor Venus have moons, and Mars's 2 companions, Phobos and Deimos, the god of war's attendants, the spirits of fear and panic, are not considered true moons but captured asteroids. Phobos is in an unstable, decaying orbit, spiraling towards the red planet as it loses momentum. Calculations show that in about 40 million years, Phobos will begin brushing through the upper reaches of the Martian atmosphere and, from there, will fall out of the sky. A similar impact on Earth would trigger mass extinction. In an odd coincidence, the Moon appears almost as big as the Sun. This means that the Earth is the only place in the entire solar system to watch a total solar eclipse, with the characteristic thin ring of fire around the edge of the Moon, where the Sun's upper atmosphere is just visible. If the Moon is crucial for complex life, its importance will be its tidal effect on Earth. For a Moon to draw significant tides, compared to the effect of the Sun's gravity, it must have an apparent size similar to, or larger than, the Sun's. The main reason for Earth's large moon's importance is its gravity stabilizes the planet's spin. The Earth rotates around its pole-to-pole axis, causing day and night cycles. This axis is not perpendicular to Earth's orbit, but tilts by 24 degrees. The gravitational tug-of-war affects the Sun and the giant gas planets Jupiter and Saturn, which form an inclination angle. The nearby Moon overpowers these other influences and keeps the Earth's tilt much more stable. Evidence indicates that Mars, with its current tilt akin to Earth's, once experienced a 60-degree slant. Without the Moon, Earth's lean would sway chaotically. Calculations have shown that it could approach an incline of 85 degrees. We know little about the effects of such a slant. We ran computer simulations of the climate without the Moon's influence, gradually toppling the entire planet head over heels until the poles were almost horizontal. The poles would receive more sunlight than the equator over a year, potentially reversing the climatic conditions in these 2 regions. It is not clear whether a swaying planet would be significantly more prone to runaway climatic processes. Complex land life, like Earth's, would probably be impossible, but we do not know whether it would matter for organisms to develop deep underground, for example. Some claim that a large gas giant planet in the outer solar system is necessary for life to evolve on a rocky planet within the stellar HZ. Impacts with comets or asteroids are serious hazards to a planet's biosphere. The one that hit Earth 65 million years ago is almost certainly the culprit in the extinction of 75% of its species, including the dinosaurs. The premise is that the gravitational influence of a gas giant clears away stray comets, either taking the hit themselves, like the renowned collision of Comet Shoemaker-Levy 9 into the Jovian clouds in 1994, or deflecting them out of the solar system. Jupiter might be a celestial goalkeeper, protecting the inner planets from potentially devastating impacts. Experts estimate that 1,000 times more comets would have fallen Earthward without Jupiter's guardian effect. However, This presumed necessity must be more convincing than the others we have explored. Cometary impacts can be beneficial only in certain situations. With the abrupt end of the dinosaurs' reign, our shrew-sized mammalian ancestors would have had the evolutionary opportunity to spread and eventually produce our conscious and technologically able species. In the next chapter, we'll see how life on Earth would be impossible without a bombardment of brutal collisions during Earth's childhood, which delivered the oceans and all the organic molecules made in nebulae. Others have asserted that Jupiter impeded the expansion of the inner solar system. Had it not existed, the asteroid belt and Mars would have formed planets larger than Earth. A more massive Mars could have retained a thicker atmosphere and warmer surface, and may have remained habitable. Others believe that a large gas planet's gravitational disruption is critical to speeding up the formation of the inner planets from colliding rocky embryos. So, a planet must be the right size, orbit a suitable star at the correct distance, and have several other appropriate solar system features. The prospects for a galaxy teeming with life get even worse when we consider that only certain regions within a galaxy may be suitable. Our Milky Way galaxy is an immense rotating spiral of around 400 billion suns, about 100,000 light-years across. We can discount many of these stars as serious contenders for life because they are too fleeting, and many more might not have terrestrial planets within their habitable zone. As I have described, metallic elements are important for life. Cells rely on the complex chemistry of a wide range of heavy elements. The rocky terrestrial planets are essentially massive lumps of iron, silicon, and oxygen, as well as the radioactively unstable isotopes of uranium, thorium, and potassium needed as a heat source deep within the planet's interior to drive plate tectonics and volcanism. The galaxy's development has concentrated these heavier elements in the center, rather than dispersing them evenly. A metallicity gradient extends from the galactic core to the outer rim. It is believed that stars near the outer rim cannot form a family of planets or provide the basics for terrestrial life. Our Sun lies about halfway between the galactic center and its edge. The decreasing abundance of heavy elements could establish an outer boundary for the distribution of life, beyond which it is impossible to assemble planets. Although the inner edge of this galactic HZ is less well defined, scientists believe it is no coincidence that the Sun is far from our galaxy's center. Certain factors may prevent life from gaining a foothold in the inner regions, as major threats to its continuation lurk throughout the galaxy. Supernovae explosions are crucial for creating and scattering heavy elements, but their sheer violence also makes them a considerable hazard to life-bearing planets, even across the gulfs of space. Supernovae, around 30 light-years away, pose the greatest threat. This is a midpoint between the devastating effects of a very close detonation and the decreasing probability that a star will be so close to Earth when it explodes. A star exploding 30 years away would hang blindingly bright in our skies, but it wouldn't be the extra light that spelled doom to ecosystems on the surface. The detonating core of the supernova would greatly accelerate material in the expanding shell, creating a pulse of highly energetic particles. Radiation like this is spread throughout the galaxy. Cells on Earth have evolved mechanisms to repair damage caused by low levels, but a nearby supernova explosion would strike the Earth like a point-blank shotgun blast. In addition to the direct problems of high mutation rates in exposed organisms, this radiation surge would destroy the ozone layer for centuries and allow UV light from the sun to stream onto the surface, damaging the delicate molecules of life. Photosynthesis machinery is particularly sensitive to UV light. A nearby supernova could shut down photosynthesis in land and marine environments, and the ecosystems dependent on it would crumble. The long-term effects of such a close explosion could be clearer. The radiation boosts cloud formation, reflects sunlight, and triggers runaway glaciation. Alternatively, the sudden drop in photosynthesis and reduced carbon dioxide removal from the atmosphere might tip the balance the other way, increasing global temperatures. Only large and relatively rare stars, larger than about 8 sun masses, result in supernovae. Although a supernova occurs every few decades in our galaxy, the chance of Earth being within 30 light years when one goes off is very small. However, at least one close detonation has likely occurred during the history of complex animal life on Earth. Since the evolution of hard-shelled animals 550 million years ago, several major mass extinctions have occurred. We confidently attribute the event that wiped out 75% of Earth's species, including the dinosaurs, 65 million years ago, to a massive impact. However, this was a relatively subdued catastrophe among the big five mass extinctions, with no convincing explanations for many others. A supernova may have triggered at least one of the major disturbances to life on Earth. However, if this was indeed the cause of a previous mass extinction, it is unlikely that we will ever uncover the definitive evidence. The relative motion of stars means that, after swinging towards Earth and exploding, the supernova remnant could now be on the far side of the galaxy. A layer of a heavy iron isotope in the deep ocean crust is evidence of a nearby supernova explosion in the last few million years, when the first human-like ancestors were evolving. The explosion occurred approximately 100 light-years away, and we can still observe the debris drifting away in our neighborhood. The Sun's orbital path through the galaxy is relatively unpopulated with stars. The threat from a nearby supernova would greatly increase if the solar system were located in a region with a much higher star density, such as toward the galactic core. The tighter packing of stars in the inner districts of the galaxy is dangerous for a second reason. Close encounters with other stars would be much more likely, with the errant gravitational tug playing havoc with the solar system. The swarm of comets lurking in the solar system's outer reaches would undoubtedly scatter, sending some plummeting toward the inner planets. A firestorm of impacts would hit Earth, posing grave consequences for life's survival. Despite the Sun passing through Sung, a relatively uncrowded galaxy area, we are still making determinations. The Milky Way has a rotating spiral structure, much like the white foam swirling around on a fresh cup of coffee. If you watch your cappuccino carefully, you will notice that the inner regions turn much faster than those further out. Likewise, stars within our galaxy constantly overtake others on their outside. The four spiral arms do not turn into rigid formations. They're just regions where many stars are born and light up the arm before they migrate away. The Sun's orbit around the galactic core regularly takes it through one of these spiral arms about every 100 million years. Each journey takes about 10 million years, and the solar system is dangerous. The spiral arms contain dense concentrations of stars, which increases the risk of supernovas. The enormous dust clouds that birth new stars are another hazard. These nebulae, concentrated in spiral arms, span hundreds of light years. The solar wind would typically be strong enough to blow away the cloud without any ill effect, but an especially dense section of cloud would overpower the Sun's protection and flood into the solar system. The dust would gravitate towards the planets and settle in the upper atmosphere, blocking the sunlight for around 200,000 years while the solar system passes through the cloud. At the least, this could shut down photosynthesis. At worst, it could kickstart global glaciation. So the spiral arms pose three main dangers: supernovae, close shaves with other stars, and interstellar dust clouds. Calculating the Sun's path around the galaxy is challenging, but some researchers have attempted to do so, finding that several mass extinctions correspond with previous crossings of spiral arms. The Sun's orbit around the galactic core is surprisingly circular, ensuring that the solar system does not periodically plunge deeper towards the center into dangerous regions with higher densities of stars. The orbit is also uncannily close to the co-rotation cycle, where the Sun's orbit around the core is almost identical to the rotation of the spiral arm pattern, resulting in a minimal number of crossings. This is the final feature of what many astronomers call the galactic habitable zone. Just as life can only persist on a planet at a specific distance from its star, the star must orbit within a ring around the galactic core. The minimum metallicity required to form rocky planets defines the outer edge of this ring, whereas the various hazards posed by the central realm of the galaxy, such as the greater danger of close encounters with other stars and nearby supernovae, define the inner boundary. Even a solar system further out in the disk is not safe, as regular crossings of the spiral arms carry similar risks. According to Figure 6, the safest place for life is within a thin halo around the co-rotation cycle, not far from the Sun's orbit. Sun's researchers calculate that this haven contains a mere 10% of the stars in the galaxy, of which only a fraction are sunlight. Therefore, life may need to be more relaxed about where it arises. It demands a fair-sized planet, set in the Goldilocks zone around a well-behaved star and in a convenient region of the galaxy. Also, entire galaxies must be of the right kind. Metallicity is important for constructing life-capable planets. Only a portion of our spiral galaxy is suitably metallic. Estimates suggest that 80% of stars in the visible universe reside in galaxies less luminous than our own, indicating that most stars are relatively metal-poor and may not be able to form planetary families. Within our local group, including the Milky Way, Andromeda, and 24 other galaxies, most are either blob-like or irregular and metal-poor. These galaxies also feature stars with much more erratic orbits that regularly plunge towards the dense galactic core, carrying all the associated dangers. Our Sun may stand out from most other galaxies within the Milky Way. These conclusions heavily depend on the range of conditions that life might be able to survive, conditions that researchers are actively working to understand. Certain issues I have discussed, such as the greater threat of a close-quarters supernova nearer the galactic center, would not prevent life from arising, but may present a hazard to its persistence, particularly the persistence of more complex life. Based on only a single data point, the requirements described in this chapter provide the most favorable conditions for life to develop and survive. In later chapters, I shall relax these constraints and determine what other galactic real estate might suit. The spark ignited interest in the possibility of life in other worlds, and it was the discovery of so-called extremophile life on Earth, organisms thriving in hostile environments previously assumed to be absolutely sterile. Interest in this branch of microbiology has grown enormously since the 1990s. Now it feels as if we invariably find life flourishing in any location we care to check. Life, once started, seems to have become utterly irrepressible, adapting to fill almost any damp niche. There is evidence for liquid water on many planets and moons within the solar system. Indeed, it ought to be abundant on any planet at an appropriate distance from its star. So these discoveries at the extremes of our world are tremendously important. Three main parameters affect the functioning of cellular biology: temperature, acidity, and salinity. Different organisms can survive over different ranges of these conditions. We can combine these ranges of tolerances for all cells and plot them on a three-dimensional graph to illustrate the full extent of life on Earth. This describes the survival envelope of life as we know it, from acidic cold water in one corner to alkaline hot brine in the opposite. The exciting realization is that parts of this bulgy cloud overlap with the conditions we believe exist in other worlds. Certain terrestrial life would be perfectly content in extraterrestrial settings regarding temperature, acidity, and salinity. We'll take each of these in turn, explaining what environments such extremophiles cope with, as well as a little about the evolutionary adaptations that ensure their survival. Because of the eukaryotic cell's complexity and sensitivity to physical and chemical extremes, only prokaryotes populate much of the outer regions of the survival envelope. However, we should remember that extremophiles are extreme only regarding other cells' capabilities. There is no standard environment on Earth, and our understanding of the conditions under which life initially emerged is limited. Extremity is different for everyone. From the point of view of a bacterium living in the intense heat and pressure of deep underground, to the cells struggling to survive in the harsh cold and low-pressure environment of the surface, which is full of UV radiation and corrosive oxygen. These environments are seen as evolutionary oddities. Water freezing and boiling into vapor set the absolute limits of temperature survivability. Active life requires liquid water. However, other factors modify water's freezing and boiling points away from the 0 degrees C and 100 degrees C we are familiar with. Pockets of briny water within solid ice can remain liquid at 20 degrees, and water in the deepest parts of the ocean only starts boiling at 400 degrees, similar to a pressure cooker. Surviving very low temperatures is often fine. For instance, fertility clinics regularly store eggs in liquid nitrogen at around 200 degrees C, allowing many organisms to freeze and reanimate. Important factors, however, limit how long a cell can remain active and grow. These cold-loving bacteria are identified as psychrophiles, as shown in the table below. The primary challenge at low temperatures is the rigidity of the cell's proteins, especially the enzymes. The catalytic function of enzymes depends on their ability to change shape, but the colder it is, the less thermal vibration helps them flex. Cells adapted to near-freezing temperatures have evolved enzymes with much looser bonds, which means they remain supple even at very low temperatures, and the cell can keep its metabolism going. Similar factors also enable their cell membranes to remain significantly more mobile. The danger facing such cells is that these looser proteins denature if the temperature rises too far above their optimum, just like the frying egg. Cold-adapted bacteria effectively start cooking at just 20 degrees C, and if the misshapen proteins don't kill the organism, the spilling out of the cell contents due to membrane ruptures certainly will. Psychrophiles are at home in Antarctic waters and deep-sea sediments. Certain psychrophiles are active within Antarctic ice at 20 degrees Celsius. As the seawater freezes, the salt becomes increasingly concentrated in small pockets, preventing the water from solidifying and producing a labyrinth of interconnected channels and pores. Such low temperatures result in prolonged reproduction times of up to 6 months, compared with bacteria living within the human gut, which divide every 20 minutes. Much of the Earth's surface biosphere is cold. More than 90% of ocean water is below 5 degrees C, so organisms adapted to the high pressures of deep water, barophiles, also tend to be psychrophilic. Heat-loving organisms, thermophiles, face the opposite difficulties. They have evolved enzymes with extra bonds to stop them from shaking apart at high temperatures, as well as membranes with reduced fluidity. Although higher temperatures speed up chemical reactions, thermophiles have an advantage because many organic molecules decompose above 150 degrees Celsius. An archaeal chemoautotroph has set the record by being the only organism to survive above 121 degrees, setting an absolute upper limit on survivability. The majority of extremely heat-tolerant organisms, hyperthermophiles, are archaea. Eukaryotes are comparative wimps. A few algae and fungi can cope up to about 60 degrees C, and the Saharan desert can forage under the midday sun at 55 degrees C. Chlorophyll degrades at 75 degrees C, so very little photosynthesis happens in hot pools. Another issue is that gases such as oxygen and carbon dioxide have a lower solubility in hotter water, causing many hyperthermophilic cells to become anaerobic. Environments such as the flanks of volcanoes, geysers, and geothermal hot springs typically host thermophils. A lot of reduced chemicals come from deep inside the Earth. Because of this, many thermophils are chemoautotrophic, which means they use oxygen, nitrate, ferrous iron, or reduced sulfur compounds to react with other chemicals. Extracting energy by oxidizing sulfur compounds has one fairly nasty side effect. These reactions produce sulfuric acid, which often makes the geothermal waters very acidic. The volcanic hot pools in Yellowstone National Park, USA, contain sulfurous gases that can be extremely acidic. The edges of these pools often display bands of vivid colors, reflecting the regions of minerals in various redox states created by different thermophilic organisms, with the water gradually becoming hotter, more reduced, and less acidic as its depth increases. Therefore, the thermophiles nearer the water's surface must also be adept at handling high acidity. The pH scale rates acidity by measuring the concentration of H+ ions, protons, in the solution. A low pH signifies an acidic solution, a high pH is alkaline, neutral is pH 7. Cells use concentrations of protons as a fundamental mechanism to transform energy. In fact, many organic molecules, including amino acids, nucleic acids, and numerous small molecules involved in metabolism, can lower pH. Many acidophiles can tolerate as low as pH 2, about the acidity of lemon juice. Some acidophilic species were first discovered when they spoiled tinned fruit. Others are common in hydrothermal pools or water running out of metal mines with sulfur ore. The most extreme can survive at pH 0, 100 times more acidic than lemon juice. When combined with high temperatures, such high acidity is extremely destructive to cellular molecules. So most acidophilic organisms can only tolerate a relatively cool 60 degrees. The stability of their proteins. Different kinds of bonds hold the exact three-dimensional structure of a protein strand together, and changes in the distribution of charges, such as an increase in acidity, can greatly disrupt these bonds. Acidophiles protect their proteins by including more amino acids with neutral side groups and actively keeping the inside of their cells as close to neutral as possible. They do this by pumping out protons as they leak across the membrane, or turning them to their advantage. For example, certain organisms oxidize iron outside the cell and transport the released electron inside to bond together oxygen and hydrogen ions, and create water. As a result, the cell removes protons, allowing those from outside to flow inward through ATP synthase and generate energy. At the other end of the spectrum, alkaliphiles survive in very alkaline waters, such as the soda lakes of the western USA and Lake Magadi in Kenya. These organisms tolerate conditions up to pH 11, equivalent to household ammonia. Under these circumstances, the concentrations of hydrogen ions are very low, and cells have trouble using ATP synthase to generate power. Other essential ions, such as magnesium and calcium, precipitate out of the water as salts and are available only at very low levels. The alkaliphiles cope by actively pumping in these ions and exporting others to keep their interior near neutrality. Various organisms can thrive in pure water, but the other end of the scale is much more problematic. Salt-loving halophiles must control osmotic balance, the difference in water concentration between the inside and outside of the cell. Osmosis is the process by which water moves into regions with the highest concentration of solutes from most to least diluted. This means that cells living in a very salty environment risk losing all their internal water to the outside. They combat this by pumping out sodium and keeping their inside contracted using benign chemicals, acids or glycerol. Thus they avoid the problem of having a very salty cytoplasm, which would disrupt and possibly denature proteins. High evaporation rates from lakes like the Dead Sea, or the trapping of salty water in ocean basins by less dense water above, create hypersaline environments. High salinity is one of the less demanding physical constraints. Even some higher organisms can survive the extremes. We often observe ecosystems of brine shrimps feeding on dense algal blooms in brine lakes. But as the water evaporates and the salinity approaches saturation point, where no more salt can dissolve, at 35% dissolved salt, 10 times more concentrated than seawater, only the halophilic archaea remain. Although discussions on extremophiles often focus on only the most impressive instances in the bacteria and archaea, there are plenty of astounding examples among eukaryotes. These are relevant to the possibility of complex ecosystems arising on alien worlds. True, eukaryotic cells cannot manage long periods at anything warmer than about 60 degrees, the temperature of a cup of tea, but many higher organisms cope with freezing conditions. The wonderfully named grillo-blatted insects scavenge over the ice sheets on mountaintops for animals killed by the cold. They are more commonly referred to as icebergs or rock crawlers, and they are active well below freezing point. If you pick one up, the warmth of your hand is enough to kill it. Because of the low temperatures, icebergs grow and develop slowly, taking up to 7 years to complete a generation. When the ground around it remains frozen solid, the American skunk cabbage can thrive in sub-zero temperatures. It accomplishes this partly by using the proton gradient established by respiration, not to make ATP, but directly to generate heat to thaw the surrounding soil. Perhaps most remarkable of all complex eukaryotes are the tardigrades, or water bears. Despite superficial similarities to arthropods like lobsters, these microscopic creatures, measuring less than a millimeter from head to tail and equipped with 8 stubby legs, are considered unique enough to warrant their own group. They are commonly found on damp, mossy patches almost anywhere, from the tropics to the poles. When the going gets tough, tardigrades transform into a barrel-shaped tun state, and they can hibernate like this indefinitely. In their tun state, tardigrades can withstand a wide range of hazards, such as temperatures ranging from 253 degrees C to 150 degrees C, intense X-rays, extremely high pressures, and even a vacuum. As true survivors of the animal world, they serve as positive models for the complex life that may exist elsewhere. In addition to identifying the limits of survivability on Earth, seeing just how far the envelope of life extends, astronomologists are keen to study spots on our planet that are thought to closely resemble known extraterrestrial environments. We'll look at 3 of these in detail. First, one is considered a beneficial analogy for life on Mars. Antarctica is the coldest place on Earth and contains some of its driest regions. As the winds blow across the southern continent, shaped like an upturned bowl, they push the frigid air, which can hold little moisture, to higher altitudes, further drying it out. These extremely desiccating winds blow across the top valleys, creating huge, barren, ice-free rock areas. There is less than half a centimeter of precipitation a year, and even this meager amount remains frozen for much of the time, inaccessible to life. During the long, bleak winter, the temperature in these dry valleys plummets to 40 degrees Celsius, soaring to a balmy 1 degree Celsius in summer, which lasts only a fortnight. Animals that become stranded in these valleys inevitably die and are instantly freeze-dried. Researchers have discovered and preserved the bodies of some seals for thousands of years. When Robert Scott explored them in 1903, he described them as the valleys of the dead because he could see no signs of life. But if you look closely enough, you find living organisms, tiny populations of nematodes and tardigrades feeding on bacteria and detritus, thawed out for the all-too-brief summer. These organisms do not crawl over the open ground. Rather, they live in tiny crevices between the grains of boulders scattered across the desolation. The rocks conceal entire ecosystems of organisms, a phenomenon known as cryptoendoliths. Life is only possible because these cracks maintain a much more pleasant microclimate than the freezing air outside. The sun's meager heat warms the rocks, and the cracks trap the melted ice and protect the inhabitants from the brutal wind. The thin cracks provide another crucial service for the photosynthetic lichen and bacteria that support these miniature ecosystems. Because of the hole in the ozone layer, Antarctica has one of the highest levels of UV on the planet. Radiation that inhibits chlorophyll and damages proteins and DNA. Buried beneath just a few millimeters of rock, Antarctica blocks much of this lethal UV light. Yet the translucent stone acts as a very effective filter, allowing enough visible light to drive photosynthesis. On Mars's surface, such endolithic habitats exist. Even on its equator, Mars's ambient temperature is frigid for most of the year. But cracks in rocks could absorb enough warmth to collect pools of meltwater and support life for short periods during the Martian summer. Mars is unprotected from UV radiation without a thick atmosphere or ozone shield. The DNA damaging effect is 400 times greater than on Earth. A thin layer of translucent rock could absorb UV light, providing similar filtering effects to those of dry valleys and allowing photosynthesis to continue. However, Martian surface life still faces significant challenges. First, the current atmospheric conditions on Mars make liquid water unfeasible due to its heat and humidity. And second, the solar UV radiation is believed to have severely oxidized the exposed Martian ground, destroying any accumulated organic molecules. It is challenging to imagine even endolithic habitats providing the basic requirements for life. But Mars has not always been this unpleasant. In Chapter 5, I shall discuss the possibility of surface life in ancient Martian times and the chance that it thrives, even today, deep underground. One mechanism for creating crevices in rocks is likely widespread in solar systems throughout the galaxy. Meteorite and cometary strikes are commonly thought to be agents of destruction, but the immense shock waves generated by such impacts fracture nearby rocks, opening up an enormous number of potential endolithic habitats. The Horton crater, for example, lies beneath the thick ice of Devon Island, the largest uninhabited island on Earth, in the far north of the Canadian Arctic. An impact excavated this 24-kilometer-wide pit about 20 million years ago, burying it beneath a thick ice sheet. A ring of shocked volcanic rocks around the crater is 25 times more porous than elsewhere. Photosynthetic organisms inhabit these cracks, thriving within their clement microenvironment for a few months a year. Impacts have broken apart the rubble that litters the Martian surface, creating many similar endolithic habitats. Equally important is the possibility that the first cells to colonize Earth's land masses used these prime residential opportunities. The early solar system's impact rate likely extensively fractured the rocks of the first continents. There is another habitat resulting from a meteorite hit that may turn out to have enormous astrobiological significance in freezing environments. The impact creates a wide area of shocked rocks and a focal point of immense heat, originating from the strike's energy and the rising of deeper, hotter crust to the surface, which melts the surrounding ice into a geothermal lake. Bacteria rising from below or arriving on the wind quickly colonize this oasis of warm water in a land of permafrost. For example, calculations show that the Horton crater maintained a pool of water at temperatures exceeding 50 degrees Celsius for thousands of years. At the tail end of the heavy bombardment, the larger impacts would have created surface hydrothermal environments capable of persisting for many millions of years, time enough to be significant in the evolution of life. Lake water percolates into the hot crust beneath and seeps back out again, rich in the dissolved minerals that can support colonies of chemoautotrophs, like the hydrothermal vents we'll visit in the next section. Over time, the stored heat gradually escapes and the lake freezes, but the water beneath this insulating cap remains liquid for many more years. As the energy gradient it depends on dissipates, the potential for life decreases, and the ecosystem winds down to a final collapse. The southern hemisphere of Mars is heavily cratered and thought to contain much subsurface water. Hydrothermal systems could have played a significant role in astrobiology on the early red planet, facilitating the formation of new colonies and niches. Scientists have discovered craters in the polar regions of Mars that still hold frozen water lakes. Scientists are yet to determine whether these were ever geothermal pools capable of supporting life, but the possibility is intriguing. The freezing of the lake water could have preserved cells within it. Hopefully, future probes will dig samples from such ice and thaw out trapped inhabitants to revive them. When the Voyager space probes discovered erupting volcanoes, new moons, and rings in the Jupiter system in the late 1970s, they also discovered something extraordinary in the dark depths of Earth's oceans. Researchers working in submersibles were familiar with barophilic life that coped with the crushing pressures of the ocean bed. Researchers have even discovered barophiles at the bottom of the Mariana Trench, the deepest point of the Earth's crust, where the pressure is over 1,000 times greater than at sea level. Although photosynthesis is impossible in these pitch-dark depths, animals are home on the deep abyssal plains, subsisting on the organic detritus that snows down from the productive sunlit regions far above. Great herds of sea cucumbers, a translucent tube-shaped animal related to the starfish, occasionally travel across the ocean floor, churning up sediment clouds like buffalo stampedes on the savannah. However, food is scarce at these depths, which, combined with the low temperatures, results in low animal populations and rapid growth rates. Until 1977, researchers believed this to be true for the entire ocean bed, but they discovered an astonishing exception that revolutionized biology. In the mid-Pacific, a long scar runs across the planet's face, forming a spreading center where fresh magma pushes up to form a new oceanic crust and forces apart two tectonic plates. Along this ridge, 2.5 kilometers beneath the waves around the Galapagos Islands, scientists crammed into a tiny submersible and discovered teeming oases of life in the cold, dark desert. Water seeps through the cracked crust and comes into contact with the hot magma chamber beneath the ridge, superheating and leaching minerals out of the surrounding rock, particularly iron and sulfur compounds, until it becomes saturated. Localized spots on the ocean floor, known as hydrothermal vents, force the water back up. Only the deep water's crushing pressure prevents the emerging water from boiling, but it cools rapidly as it mixes with the frigid ocean around it. Its carrying capacity drops, and many dissolved minerals instantly precipitate out. This results in a massive cloudy plume of dark particles, leading to the nickname black smokers for these vents. Some of this mineral material accumulates around the vent, building up tall chimneys that funnel the hot water out at the top. These occasionally collapse under their own weight, but some grow to truly monstrous sizes, such as the Godzilla black smoker off the coast of Washington state, which is higher than a 16-story skyscraper. It wasn't the area's geology that surprised the scientists so much as the wealth of life thriving there. Pale crabs and shrimp scuttled around the chimneys in the cooler water, feeding off giant dinner plate-sized clams, mollusks estimated to grow 300 times more quickly than their relatives on the abyssal plains, a testament to the abundance of energy pouring out of the vents. Most astounding were the tube worms, alien-looking animals with blood-red heads, up to 1.5 meters long and completely new to science. Such vent ecosystems are extraordinarily ancient. As we will explore in Chapter 4, life may have arisen around hydrothermal vents on the primordial Earth. Only when we brought some tube worms and clams to the surface for closer inspection did the mystery surrounding them deepen. Researchers discovered that they completely lacked both mouths and digestive systems. How could they feed in order to stay alive? It took another 4 years to discover their secret, symbiosis, a particularly close association between two organisms. The vent jet is laden with reduced ions from within the crust, which support a large population of chemoautotrophs around the plume's edges. These organisms must also be thermophilic. The most heat-tolerant organism on Earth lives around these vents, coping with temperatures of up to 121 degrees Celsius. Many of these chemoautotrophs speed up the oxidation of reduced sulfur or iron compounds. They do this by using the redox reaction to get energy and fix the large amounts of carbon dioxide in their needed organic molecules. The tube worms survive on these chemoautotrophs, not eating them, but hosting a large colony within a specially socialized organ almost half the size of the worm. The red head of the worm functions like a gill, stuffed full of hemoglobin proteins, similar to the pigment in human blood but independently evolved, which absorb oxygen and hydrogen sulfide from the water. The chemoautotrophs receive the chemicals, carry out the redox reaction, and provide the worm with nutrients in exchange for shelter and food. There was an even bigger shock in store for the scientists. Less sunlight penetrates these ocean depths than reaches Neptune, yet they found shrimp with light-sensitive organs on their backs. What on earth could these animals be seeing? The shrimp most likely senses the faint light from the superheated vent water, avoiding scaling. But this realization presents another possibility. If animals use this dim glow, could cells photosynthesize by it? In 2005, researchers collected photosynthetic bacteria from vent water samples. The proof is still lacking, but if organisms can survive on geothermal light alone, this opens up a completely unexpected energy source for astrobiology. Seafloor spreading ridges worldwide now host similar vents, but only those in the Pacific are home to giant tube worms and clams. Each vent has a particular chemical composition, water temperature, and flow rate. Some gently mix with the surrounding seawater, so don't produce concentrated plumes of pre-precipitation. In terms of possibilities for astrobiology, hydrothermal vents may represent an almost universal mode of life. Thermophilic chemoautotrophs can support an ecosystem of bacteria and animals on inorganic redox reactions far from the sun. However, even this deep life is indirectly dependent on photosynthesis, as most of the dissolved oxidizing power in the sea comes from plants in the sunlit waters above. Black smokers can run entirely on abiotic oxidants, as the first cells are believed to have done, but the lower energy availability would severely limit the size of the ecosystem. Any planet or moon with a watery ocean and source of internal heat could provide such a habitat, with subsurface ecosystems at the interface between the hot, reduced crust and the cold, oxidized water. We'll explore this possibility further when considering Europa, the ice moon of Jupiter. Photosynthesis on Earth still partially supports hydrothermal vents, a promising model for extraterrestrial life. However, a few examples of life are isolated, completely independent of sunlight, and self-reliant on energy and organic substances. These ecosystems, powered by chemoautotrophs, provide the best general model on Earth for life elsewhere. One is in a thick layer of rock, lying beneath what is now the Columbia River Basin in Washington State, USA. Around 10 million years ago, a series of large floods of volcanic lava formed this basaltic rock across the surface. Basaltic rock is rich in reduced iron and reacts slowly with aquifer water to release hydrogen. Chemoautotrophic archaea living a kilometer deep within derive all their energy by oxidizing this hydrogen, passing the freed electron to dissolved carbon dioxide and fixing it. These organisms, known as methanogens, produce water and methane gas from this redox reaction. It is believed that these anaerobic methanogens and the heterotrophs they consume do not require oxygen or the organic molecules produced by photosynthesis above. They are part of something called subsurface lithotrophic microbial ecosystems, slimes. Another advantage for these communities is that they do not require active geothermal heating hotspots like the black smokers. They need a slab of reduced volcanic rock. How deep life extends into Earth's crust is another topic of intense research. Miles underground, there are still minute pores and fractures in the rock, which is filled with water and large enough for bacteria. The furthest scientists have ever poked into the Earth's crust and sampled is 5.3 kilometers down a borehole in Sweden. Even at the bottom, at a depth where the rock temperature is greater than 70 degrees Celsius, there are a variety of active heterotrophic bacteria. There is likely no survival limit to depth, so long as the ambient temperature does not become lethal. A thermophile can withstand temperatures as high as 121 degrees Celsius. Depending on the thickness of the crust, this point can reach different depths, but in some sedimentary rock formations, it can reach as deep as 10 kilometers. The cells of a deep slime are extremely slow-growing and live at low density. At some sites, only a few thousand cells per gram of sediment, about a millionth that of fertile topsoil. However, considering the vast volume of the upper crust on Earth, this could still constitute a staggering mass of living material. Some researchers have estimated that the deep, hot biosphere exceeds the total sum of all surface life. These subterranean fissures serve as ideal habitats in many ways, providing a steady, albeit slightly limiting, flow of chemical energy without UV or energetic cosmic radiation, and shielding the cells from all but the most devastating catastrophes above. The deep, hot biosphere would be unfazed by a large impact or global glaciation. Basaltic rock, water, and dissolved carbon dioxide are considered common substances on terrestrial planets or moons capable of volcanism, and so many believe the slime model to be a surefire method of supporting alien life. This presupposes life's evolution. The possibility of its emergence in such an environment as opposed to mere colonization remains undetermined. Such conditions are believed to exist far underground on Mars. Impacts have heavily fractured the top layer of Martian rock, and its subsurface is believed to be filled with ice-laden pores. This permafrost would melt around geothermal hotspots and react with the basaltic rock. Archaea, that metabolize hydrogen on Earth, releases methane, and recent discoveries on Mars have revealed intriguing methane plumes. Could this be the signature of a deep slime layer on the red planet? Earth's biosphere is a vanishingly thin film across the very surface, sandwiched between the rock-melting heat of the mantle below and the cold void of space above. We have found cells roughly 5 kilometers deep into the crust and around 40 kilometers into the thinning atmosphere. But even within the habitats of this one planet, organisms cope with a staggering range of hazardous conditions. Extremophiles thrive in boiling acid, freezing brines, and deep underground, living off naught but gases bubbling from the rocks. However, how extreme is it? How hostile an environment can life tolerate while still growing? Could life cope even with exposure to outer space? Consider the possibility of cell transfer between planets. People first widely discussed the theory of panspermia, which literally means seeds everywhere, in the early 1900s. People believed that their star's radiation pressure could disperse hardy microscopic cells, such as bacterial spores, throughout the galaxy. The original theory fell out of favor because unprotected cells would never survive the time required to travel between stars. However, the essence of the idea, adapted with the notion that cells could be transported between planets and moons aboard meteorites, has recently been resurrected and is rapidly gathering encouraging evidence. There are essentially 3 hurdles that bacteria must leap to survive the journey between 2 planets. A fragment of the surrounding rock ejected them from their home planet. The meteorite's transit exposes it to a hostile space environment. The atmosphere is where an impact re-enters the destination planet. Calculations and experiments at each stage generally show no acute problem with the prospect of panspermia. Originally, scientists believed that launching living cells from a planet would unlikely survive. An impact must be very energetic to throw up fragments of the target surface at sufficient speed to punch through the atmosphere, escape the world's gravitational pull entirely, and enter interplanetary space. Such a violent strike would dump enormous heat into the ground, flash boil any water, and send an immense pressure pulse rippling through the solid rock, shaking and crushing all nearby life. The acceleration required to propel the planet away should be sufficient to flatten any cells. This is certainly true for the ground closest to the impact point, but mathematical analysis has found an intriguing surrounding region, the spallation zone. Pressure pulses propagating through the crust refract to the surface, where they meet a more direct wave heading sideways away from the impact site. The two merge and, in certain regions, destructively interfere, cancelling out much of the intense pressure. You can see the same process when throwing a stone into a pond. In some places, 2 crests coincide and combine together. In others, a crest and a trough meet to produce flat water. The spallation zone propels lumps of rock upwards at high velocity without causing immense shock. This process is particularly efficient if the impacting body plows into the ground at a glancing angle. Much of the rock above the strike explodes outwards relatively unharmed, Yet a single impact can hurl millions of tons of surface rock into space. The brief heat pulse does not penetrate deep into the boulders, protecting bacteria insulated in the center from cooking. This process has blasted off several lumps of Martian rock from their homes on Earth. Upon analysis, we discovered that these lumps of Martian rock experienced relatively minor peak pressures, and their interiors remained below 100 degrees C. Lab experiments have addressed the question of whether living cells could actually survive these conditions. Scientists have soaked porous ceramic pellets with bacteria and fired them into a rigid target at extremely high speeds, reaching up to 12,000 miles per hour. During the crushing impact, the cells are subject to huge deceleration forces. But between 1 in 10,000 and 1 in a million, bacteria recover and continue growing. This demonstrates that a substantial portion of the inhabitants of surface rocks can endure the harrowing experience of a forcible spacewalk. Once off their homeworld, things get decidedly worse for the bacterial stowaways. The environment in space is extremely hostile, far removed from any terrestrial niche one could imagine. It is extremely cold, a near-perfect vacuum, bathed in radiation and free fall. Weightlessness is not a major problem for a cell's function, and many organisms survive the deep freeze without harm. However, radiation is likely to be a major limiting factor. UV can kill an unprotected cell within minutes, a hazard exacerbated by the extremely desiccating effect of vacuum. However, a filter of just a few millimeters of rock provides sufficient protection for Antarctic endoliths. The background radiation of highly energetic particles surging from solar flares and supernovae throughout the galaxy is much more of an issue. The Earth's magnetic field and thick atmosphere shelter surface life from this radiation. But bacteria within a meteorite would need several meters of solid rock for complete shielding. These energetic particle cascades steadily degrade the delicate molecules in a dormant cell, setting a time limit for the meteorite to land on another planet before all the cells perish. The vacuum of space will inflict extreme destruction on their molecular structure. The carbohydrates, proteins, and DNA within a cell begin irreversibly to cross-link, undergoing the same chemical reactions as a steak browning on the grill. We tested all of these factors on live cells launched into orbit and exposed to the space environment. As expected, the cells provided no protection against UV radiation and died almost instantly. Yet a significant number managed to survive the 6-year mission with minimal shielding, aided only by the few layers of dead cells above. Could a population of cells, sufficiently protected from the radiation environment, remain in suspended animation long enough for their meteorite to crash land on another planet? To transfer between Mars and Earth, it would typically take 15 million years for a meteorite's orbit to gradually distort until it begins crossing the Earth's path. Some simulations show a tiny fraction of rocks are ejected on a much more direct trajectory, being delivered first class within just several thousand years. But, in general, for panspermia to be plausible, bacteria must be able to survive in space for many millennia. Lying dormant is something bacteria are excellent at. Many bacteria, such as those common in soil, form resilient spores when conditions turn too cold or dry, or food runs out. A hard outer casing protects this desiccated capsule, while special proteins stabilize its DNA. Such spores are very resistant to environmental assaults, including radiation, vacuum, temperature extremes, and hazardous chemicals, and they simply reawaken once their surroundings become favorable again. Other prokaryotes and some eukaryotes, such as the tardigrade, can endure extended periods in a dormant state that halts growth and metabolism. Just how long some cells have been dormant yet remained viable is astounding. Egyptian tombs have revived dry spores after 3,500 years, psychrophiles after freezing in ice for around 500,000 years, and bacteria from 40-million-year-old amber. However, a sample of halophiles, isolated from a large salt deposit deep beneath what is now the North Sea, is the longest sleeping of all. When Europe positioned itself over the equator, a lake evaporated, resulting in these deposits. Bacteria have been extracted from the ancient fluid trapped within the crystals and shown to reawake. According to the hotly debated claim, if the cells were as old as the rock, they would have been dormant for a staggering 250 million years. All in all, dormant cells or spores buried right in the core of a reasonably sized rock are likely to survive in space for extended periods. The actual arrival is the final obstacle. In many ways, the hazards of safe delivery to a new planet mirror those of departure. If the planet has a thick atmosphere, air friction will significantly heat the surface of the plummeting rock, forming a shooting star. However, for the few seconds of re-entry, the larger boulders that have already shielded their cargo of cells from radiation will also shield them from high temperatures. The bulk of the meteorite safely insulates the bacteria deep inside, even if the outside melts into a charred crust. To test this, we embedded bacteria-soaked pieces of volcanic and sedimentary rocks in a satellite's heat shield. Unfortunately, the biological samples had fallen off, making it impossible to analyze them for survivors. Another attempt is planned soon. Planetfall is a sharp deceleration, which sends a shockwave racing through the meteorite as it hits the ground. We've already seen that an appreciable fraction of cells survive such experiments, particularly if the meteorite falls into ice or the ocean. Smaller grains of rock might provide only limited long-term shelter from radiation, but conversely, they would tumble through the atmosphere without significant heating and land softly on the ground. Calculations show that the exchange of rocks between planets is actually quite common. For instance, since the solar system's formation, Mars has blasted off over 1 billion fragments smaller than a few meters across without excessive heating or shock. About 5% of these meteorites arrived on Earth within 8 million years. We have found about 40 of these interplanetary immigrants. In the chapter on Mars, I will recount the story of a particularly notorious one, known by the rather bland name ALHH 4001. The reverse traffic, Earth-Mars, would have been about 100 times less due to our planet's stronger gravity. The high rate of impacts would have battered the inner planets, causing a busy cross-fertilization of cells if life had emerged very early in the solar system on Mars, perhaps Venus, and even on Earth. Bacteria within meteorites that land on ice rather than rock have a much higher survival rate, so terrestrial colonization of the moons of gas giants is worth considering. Although throwing a rock that far into the outer solar system is tricky, exchanging life in this direction is perhaps 1 million times less likely. All 3 stages of panspermia, ejection, transfer, and arrival, offer very low survival rates for the organisms aboard the meteorite. Perhaps only a 1 in 100 million chance exists for each organism to survive the voyage. Odds more commonly found in lottery jackpots. However, each lump of rock that leaves its homeworld could potentially contain billions upon billions of tickets to this lottery. Litho-autotrophic cells live in the pores of deep basalt rocks at densities of up to 100 million per kilogram, and the abundance of spores in surface rock is 1,000 times higher. Even a small chunk of Earth's crust ejected into space would teem with life. Accretion, the birth of a star and its planets. Stellar furnaces forge the heavy elements required for life, then redistribute them into interstellar nebulae to construct suitable rocky planets and complex polymers. But how are these enormous clouds of gas and dust molded into stars and planets? Exploding stars are once again the key. The shock wave from a nearby supernova explosion compresses a nebula region enough for it to begin to collapse under its own gravity. The contracting gas, eddying only gently, rotates faster and faster to conserve momentum, just as an ice skater spins more rapidly when she draws in her arms. The violently swirling cloud breaks into smaller vortices, forming individual solar systems. Depending on the size of the fragments of rotating gas, they will either form an isolated solar system with a single central star, or clusters of multiple stars whirling around each other. In one go, The original explosion creates a collection of stars that sparks into light several million years after the supernova event. Our Sun, although created in the same event as many others, was born a singleton and does not orbit in a cluster of stars. As the individual vortex of turbulent gas that will form our solar system collapses, it flattens into a rotating disk. The accretion disk, a material skirt, encircles the central region that will give birth to the Sun. While only 1/100th the mass of the Sun, this disk is enough to create a family of planets, asteroids, and comets. This accretion disk stretches 10 times beyond the current orbit of the dwarf planet Pluto. 99% of the disk is gas. The remaining tiny particles of floating dust are at a density of only a few per cubic meter. The swirling dust particles stick together, growing into larger and larger lumps. Once they become large enough to exert an appreciable gravitational tug, their growth rapidly accelerates, and the rich get richer as they pull in more and more material. Within about 1,000,000 years, the dusty disk accretes into several hundred planetary embryos, somewhere between the Moon and Mars in size. The gravitational interactions amongst the swarm of orbiting bodies sometimes swing an embryo into the Sun, or catapult it out of the solar system altogether. The embryos occasionally collide in crunching and shattering impacts, forming the rocky inner planets we know today. The heat of these impacts from the decay of radioactive elements in the rock melts the interior of the growing planets, and the heavier elements, such as iron, settle into the core. However, these terrestrial planets are almost completely devoid of carbon or water, two substances considered essential for life. This is because they form in the central region of the solar nebula, inside what is known as the ice formation boundary or snowline. This inner zone, extending out about 5 astronomical units, is too warm for volatile compounds such as hydrogen, carbon monoxide, and water to incorporate into the growing dust grains. Later in the solar system's development, delivering such volatile substances to terrestrial planets is a critical process we'll examine more closely soon. The growth of embryos into full-blown terrestrial planets takes about 100 million years. Meanwhile, the gas giant planets are forming in the outer regions of the solar nebula. Beyond the snowline, temperatures in the accretion disk are below 120 degrees Celsius, and the volatile gases condense into crystals. The extra material available in these suburbs leads to the formation of much larger planetary embryos. Jupiter, for example, has a core of rocky silicates and ice about 30 times larger than Earth's. The gravitational attraction of such enormous bodies is strong enough to draw in large amounts of the nebular gas, mostly hydrogen and helium. This makes the planet more massive, pulling in extra gas and causing a snowball effect, quickly producing gigantic gas worlds. Jupiter, king of the planets, will grow 318 times heavier than Earth. Even further out in the developing solar system, there is little material available to build planets, and only small icy bodies, like Pluto or comets, form. While the accretion disk evolves from a swarm of embryos to a mature family of inner terrestrial planets and outer gas giants, the Sun develops as the nebula's central zone contracts and its density and temperature increase. After about 500,000 years, the proto-Sun becomes hot enough for the first stages of nuclear fusion to light up the inside of the cloud. The proto-Sun expands significantly, approximately 5 times its current size, but contracts as additional surrounding gases accumulate on its surface. Eventually, the density and temperature in the core surpass a critical threshold, approximately 10 million degrees Celsius, leading to the ignition of the main nuclear reaction, hydrogen fusion. Some 40 million years after its initial collapse, the Sun ends its infancy. Today, the Sun is about halfway through the stable phase of its life, steadily brightening as it approaches the inevitable endpoint of red gianthood. The Earth formed about 4.5 billion years ago, in an orbit perilously close to that of another, smaller, young planet about the size of Mars. Almost immediately, these two neighboring worlds cracked into each other. an explosive eruption ejected half of Earth's mantle into space, causing a tsunami of molten rock to race around the globe. The impact on the smaller planet was significantly more severe. The planet experienced complete destruction, with its metallic core merging with Earth's and its outer rock splattering into space. Most of this material rained back down onto the Earth's surface, but some remained in orbit. The ejected material may initially have looked like Saturn's rings, albeit composed of shards of rock rather than glittering gems of ice, circling a glowing red-hot Earth. The pieces of the two shattered planets gradually collected into a set of small moonlets, then coalesced into the single lunar body we are familiar with today. A few thousand years after this big splat, the Earth's global ocean of molten magma had cooled enough to develop a rind and solidify into a crust. Over time, probably around 4.4 billion years ago, the world became cool enough for water vapor to condense out of the air. Great oceans formed, broken only by tiny continents, an estimated 20 times less land surface than now. The high internal heat fed huge volcanoes, whose peaks emerged above the waves like the Hawaiian Islands, venting out large volumes of gas and cloaking the planet in a thick atmosphere. In the early days, the moon was much nearer to the Earth, looming in the sky twice as large as it is today, raising enormous tides over the tiny continents. Over time, this has sapped the moon's orbital energy, and it is steadily drifting backward. Tidal friction has also drained energy from the Earth's rotation, and it spins much more slowly now. Just after the moon's formation, a day lasted only 5 hours. It is possible life developed even this early, although we will never know. The planet's active surface long ago destroyed all rock from the earliest period of Earth's history. Rigid slabs segment the Earth's crust, and when these tectonic plates collide, the heat of the interior pushes one beneath the other, melting it. Above this subduction zone, volcanic activity triggers the production of granitic rocks that are significantly lighter than the original crust. Instead of subducting these new, more buoyant blocks, they merge to form the first continental crust. The oceanic crust collects water over its heavier, lower-lying rocks. The oldest rocks on Earth come from these early continents, where rivers ran over the stark bare rock, leaching out the minerals and washing into the seas to turn them salty. So, the early oceans sloshed back and forth under the powerful tidal action of the nearby moon. But if none of the terrestrial planets had any volatile substances because they formed within the snowline, where did the ocean's water come from? The answer lies in the swarm of bits and pieces left over from planet creation. The outer asteroid belt and comets formed beyond the snowline and are rich in the volatile chemicals that life requires. During the earliest stage of the solar system, a barrage of these bodies rained on the inner planets. It's unclear whether this bombardment was a tailing off after the violent formotion of the planets, or whether there was a sudden volley of incoming projectiles after a period of relative peace. Projectiles from the late formation of the gas planets Neptune and Uranus, a slight shift in Jupiter's orbit, or the passage of a nearby star through the stellar nursery that created the Sun, could have caused such a late, heavy bombardment, if the latter scenario is correct. This sharp blitz is believed to have started 4.2 billion years ago, with increasingly infrequent large impacts lasting until 3.8 billion years ago. Plate tectonics has either eroded or dragged the Earth's original surface into its interior, leaving only a pristine record of craters on the Moon. The number and size of the Moon's craters from this era, some the size of continents, are a record of the fierce blizzard of rock and ice that must have rained down on all the inner planets. This influx of comets and meteorites delivered enormous amounts of vital volatile substances to the Earth's surface. Water to fill the oceans, carbon dioxide to warm the atmosphere, and the simple organic molecules formed in the nebulae rained down like manna from heaven. The Earth still receives hundreds of tons of organic carbon annually, a rate that would have been hundreds of times higher in the early solar system. The bombardment was absolutely crucial for the development of life on Earth. Without these volatile substances delivered from beyond the snow line, no organic molecules or oceans would have ever existed on Earth's barren surface. The barrage of a late heavy bombardment may have snuffed out a life that had gained a foothold relatively quickly after the formation of the Earth and Moon. Although the vast majority of impacts were small, every now and then, the Earth stumbled into a giant lump of rock. Objects hundreds of kilometers across occasionally fell from the sky and dumped a colossal amount of energy onto the Earth's surface. Such a large asteroid would punch through the solid crust and into the fluid mantle beneath, throwing up a huge splash of molten rock that would rain back to Earth, covering the surface with hundreds of meters of solidifying debris. A thick atmosphere of rock vapor forms, trapping the heat, so the ground is a searing inferno. Under these conditions, the oceans boil bone dry in just a few months. The air remains heavy with steam for centuries, exerting a powerful greenhouse effect and baking the ground to temperatures of around 2000 degrees C, destroying any trace of life or accumulated organic molecules. Eventually, this excess heat radiates away, and the atmosphere steadily cools until droplets of liquid water fall. It begins to rain again on Earth, a continuous deluge that lasts thousands of years as the oceans refill. Such devastation may have befallen the Earth 5 or 6 times during the late heavy bombardment. After each mega impact, the oceans refilled, organic substances delivered from outer space accumulated, and perhaps replicating molecules emerged, and cells took a tentative foothold. The next cataclysmic spasm mercilessly extinguished life once more. This knockback, or impact frustration, limits the earliest time life could possibly have arisen and persisted on Earth. However, as the bombardment eased off, the impacts became less frequent and severe. The blitz's easing brought the planet relative stability, and life finally had a fighting chance. It's startling just how quickly life seems to have sprung up. The last sterilizing impact is believed to have hit Earth 4 to 3.9 billion years ago. Just 100 million years later, the first signs of life appear in the rocks. This is an incredibly short time, considering the increased complexity involved in progressing from simple organic compounds like amino acids and sugars via replicating molecules and self-sustaining metabolisms to fully integrated cells drawing energy from photosynthesis or inorganic respiration. Perhaps suspiciously short. Perhaps life didn't evolve quickly. The first evidence of life, which is highly controversial and potentially misinterpreted, didn't appear for another 300 million years. We'll delve into this debate in more detail later. One possibility is that the final planet-sterilizing impact didn't occur so recently, giving life much longer to get going. Or the last event may not have killed off all life, but left a few survivors clinging on in protected refugees, or perhaps just some complex organic molecules, giving the next genesis a head start. The panspermia theory presents a third possible explanation for the rapid appearance of life as soon as the planet became fertile. Perhaps cells didn't evolve quickly on Earth after the last impact, but arrived fully formed in a meteorite. It is unlikely that life can travel across different star systems, especially not in a mere speck of dust. Therefore, any meteorite containing life must have originated within our solar system, potentially on Mars. Mars is a smaller target than Earth, so it would have received fewer hits during the bombardment and cooled down more quickly afterwards. If Mars ever became habitable, it likely began its lifelong evolution before Earth. These cells could have traveled to Earth through a stream of meteorites, but they could only permanently colonize Earth once it became receptive following the last massive impact. At least the possibility exists that humans and all terrestrial life are Martian. Given the current lack of evidence of any present or past life on Mars, it is distinctly more probable that Mother Earth reseeded herself. A sterilizing event may have ejected life from the surface within many bits of rock, allowing it to emerge in a gap between mega impacts, return to Earth after its recovery, and repopulate the planet. However, life on Earth took time to arise. Some researchers claim that life could originate from a pool of simple organic building blocks in as little as 20 million years. Given a broth of liquid water and organic molecules, systems that can rapidly develop into replicating molecules are inevitable. We know many simple self-organizing systems. Natural selection takes over once we achieve information storage and transmission, gradually producing fitter and more complex organisms. Such a quick start for terrestrial life is very encouraging for the prospects of it arising in other worlds, especially given the prevalence of organic molecules in nebulae throughout the galaxy. Unfortunately, exactly what happened on Earth between the ceasefire of the heavy bombardment and the emergence of cells is one of its greatest mysteries. Prebiotic chemistry leaves no fossil record, making it difficult to piece together the chain of events. However, it is possible to recreate the theoretical conditions in the lab, test different environments to see which produces the best results, and consider what processes must have occurred in the emergence of life. We shall now turn our attention to this intricate detective work, before moving on to our planet's first signs of cellular life. Prebiotic chemistry. Cells have two subsystems. The first subsystem retains the blueprints for cell construction, replicating them in bulk during cell division or individually when specific new components are required. The other is the cell's metabolism, an intricate network of biochemical reactions that provide energy, as well as a rich inventory of chemicals needed to build the cell's parts and copy genetic information. Though they overlap today, both functions must have been separate at birth. First, let's explore the possible production of the organic molecules comprising metabolism and information storage polymers. In Chapter 3, I detailed the detection of simple organic molecules in the interstellar nebulae, the clouds that will eventually collapse to form new stars and planets. Among the substances known to be present, hydrogen cyanide and formaldehyde are particularly important. During the First World War, people used hydrogen cyanide as a chemical weapon, but its activities on the primordial Earth would have been much more beneficial. When dissolved in water, it reacts to produce a diversity of essential biological molecules, such as urea, amino acids, and nucleic acid bases, while formaldehyde polymerizes in slightly alkaline water to form a range of different sugars. Carbonaceous meteorites contain amino acids, sugars, nucleic acid bases, and fatty acids. This cornucopia of preformed building blocks would have rained from the sky during the heavy bombardment, mixing into a lush organic soup in the warm seas and lakes of the early Earth. Despite the destruction of most of these molecules, carbon and water were essential raw materials for life's emergence. After releasing the volatile chemicals, Earth may have employed a second organic substance production method. The young planet had a reduced atmosphere. Photosynthesis produced oxygen much later, and reduced gases like methane, carbon monoxide, ammonia, and hydrogen were the main constituents of the air. UV light from the sun and frequent lightning flashes would have provided the energy for these gases to react together. The production of larger organic molecules would have caused them to gently settle out of the atmosphere and collect in pools, similar to what space might have done. This warm water would have undergone further chemical processing, creating what is known as the primordial soup or Darwin's pond. One famous investigation, the Urey-Miller experiment, carried out in the 1950s, simulated the atmosphere and warm oceans of the early Earth. The experiment sealed a glass flask with water and a mixture of the reduced gases methane, ammonia, and hydrogen. We evaporated the water and passed it through a tube, where a discharging spark replicated the effects of lightning. We recondensed the vapors and dripped them back into the flask to complete the water cycle. We analyzed the chemical contents of the water after a week. Urey and Miller found an impressive yield of amino acids in the bottom of their flasks. Similar experiments since then have produced sugars and other organics. For a long time, this classic piece of research was considered convincing evidence that chemical synthesis in a reducing atmosphere was the origin of life. However, there is now some doubt whether the Urey-Miller experiment reproduces the conditions of primordial Earth. First, the lightning is unlikely to have been quite as vigorous as they thought. And second, complex molecules may have broken apart as they drifted down from the clouds. More importantly, we now believe that the early atmosphere was significantly less reduced than previously thought. According to UV light, widespread volcanism would have spewed out much more carbon dioxide than methane, destroying both methane and ammonia. The atmosphere was therefore much more likely to have been mainly carbon dioxide and nitrogen, with mere traces of methane and hydrogen. Repeating the Urey-Miller experiment with this mixture yields practically no results, as the synthesis of organic substances necessitates a highly reducing environment. A third mechanism for organic synthesis is currently gaining much support, and it also includes a plausible location for the origin of life: the hydrothermal vents strung along the tectonic spreading regions. These outlets release a rich supply of reduced chemicals, which would have been much more common on the hot, young Earth. Their inorganic reducing power may have fixed carbon into organic building blocks, according to the theory. The proposed redox reaction occurs between iron sulfide and hydrogen sulfide, the gas that gives rotten eggs their disgusting smell, producing hydrogen and iron disulfide. Iron disulfide, or pyrites, was commonly known to hapless 19th century prospectors as fool's gold. Oxidation of the iron releases electrons that contribute to the abundant carbon dioxide bubbling into the ocean water. Photosynthesis uses light to create reduced organic molecules that effectively fix carbon. Not only that, but the growing crystals of iron pyrite are also excellent at binding to the newly reduced organic molecules, holding them to a solid surface. This acts as a catalyst, increasing the reaction rate between simple organic substances and producing larger molecules. The black smoker chimney walls are also very porous, providing many tiny pockets where organic substances can concentrate. Small, self-sustaining reactions might have set up within these pockets, with fresh reactions diffusing in from the vent plume and waste products washing away. These rocky reaction chambers could have formed the template of the first cell, with a membrane of fatty acids replacing the open wall. Experiments simulating the conditions found in hydrothermal vents have recreated intriguing chemistry, producing long-chain carbon molecules and amino acids that polymerize into short protein strings. Some researchers believe the first metabolic networks appeared in the dark depths around hydrothermal vents, rather than in the bright primordial ponds. The role of iron in this hypothetical genesis is particularly interesting. Iron and other metals such as nickel, copper, and zinc can readily switch their redox state, either accepting or donating electrons. This makes these metals very useful for facilitating redox reactions in metabolism. More than a third of enzymes contain at least one metal atom. This means there could be a direct link between the metal ions spewing out of hydrothermal vents and the complex enzymes life uses today. As with all other ideas on life's origins, the accuracy of the black smoker theory is difficult to determine. Tectonic recycling has long since destroyed all the original oceanic crust, and lab experiments have needed to be faster to demonstrate that metabolic networks can arise in vent environments. Some researchers think the vents were a net remover of accumulated organic substances rather than a source. Hydrothermal vents circulate the ocean every 10 million years, destroying any dissolved organic substances in the superheated water. The first organic substances probably arose from a combination of sources, from above, below, and beyond, from lightning and UV light in a reducing atmosphere, from hydrothermal vents, and by delivery from outer space. There are also a variety of theories about the order of biogenesis events. Some researchers support the sequence of membrane bubbles, proteins, and genetics, whereas others favor the primacy of genetic information storage. Metabolism precedes genetics, incorporating proteins and an enclosing membrane along the way. There is emerging consensus about the construction of the first metabolic networks. A set of autocatalytic reactions is key. One of the other products of the network catalyzes each step in such reactions. Some of these products will catalyze each other within a sufficiently large pool of chemicals, thereby engaging in different reactions. Thus, these products increase in frequency, and as a wider range of reactants emerges, more potential catalysts are available to influence the others. As more molecules production increases, the catalytic interaction network intensifies, leading to the spontaneous emergence of a single self-sustaining network. This network produces more and more of the compounds in the cycle, and over time, more reactions become involved, enlarging the circuit or adding branching points. Catalytic surfaces, such as inorganic compounds or clay, are also involved. Some sections of metabolism are common to all life and must have appeared very early in prebiotic chemistry. As these ancient biochemical networks grew increasingly complex, they initiated the production of previously non-existent small organic substances and polymers, which UV light acting on vapors or inorganic reactions in black smokers did not produce. Metabolism existed before genetics, which is somewhat uncertain. However, the complexity of modern information storage molecules like RNA and DNA suggests that basic biochemical networks must have existed first to provide the necessary parts and energy to make any polymer complex enough to store and copy information reliably. The next part of the story is the emergence of these replicating polymers. Until the 1980s, there was thought to be a deep paradox in the emergence of life. DNA codes the genetic information translated into proteins, but this process requires enzymes, energy, and precursor molecules supplied by a metabolic network. Modern metabolic networks are fabulously elaborate and cannot operate without the catalytic action of enzymes, themselves coded by DNA. Therefore, DNA cannot function without metabolism, and metabolism cannot function without DNA. There is a deep chicken and egg problem at the beginning of life. Discovering that RNA molecules, already known to store information within the cell, also had catalytic properties, they could assist certain chemical reactions, like enzymes, resolved this paradox. These ribozymes bridge the realms of biochemical networks and replicating information polymers. They are believed to have dominated the planet for a period known as the RNA world. The first ribozymes were no more than short chains of nucleic acids folded into a limited three-dimensional structure that allowed them weakly to accelerate biochemical reactions. Various ribozymes coexisted, some catalyzing more useful reactions than others within the primordial metabolic networks. We do not yet know the full range of possible ribozyme reactions, but they may include a variety of redox reactions and the synthesis of sugar polymers. The current research goal is to experimentally demonstrate that they can catalyze the production of their building blocks and nucleotides, or harness redox reactions to link amino acids into a protein. However, at some stage, ribozymes must have emerged that catalyzed the most important reaction, synthesizing other strands in their own likeness, and thus becoming capable of self-replication. Once they learned this trick, RNA could drive metabolism and reproduce itself, thereby initiating the powerful engine of natural selection. Different varieties would have engaged in fierce rivalry, eventually outcompeting the less effective ribozymes. This first occurrence of evolution gradually produced ribozymes that could replicate faster, more accurately, or cooperate for their mutual benefit. Likely, groups of different ribozymes came together, with one ribozyme producing nucleotides, another fatty acid to make the membrane that holds them bigger, and a third helping to copy itself and the other two. When the fatty acid bubble swells to an unstable size, it splits into two, the basis of cell division. RNA replications would be prone to errors, introducing mutations into the ribozymes they copied. These new variants might catalyze different reaction steps, improving the range of precursors the system could use. A second revolution occurred: the appearance of RNA molecules containing a code that did not describe how to build themselves, but how to build a protein. Other ribozymes read this code and strung together the required amino acids. These genetically encoded proteins came under evolutionary pressure, adapting them into increasingly effective catalysts. Thus, the RNA world developed a more productive workforce, a set of enzymes able to take over the task of running the metabolism and replicating the genes, while the RNA remained responsible for holding the genetic information. At some point in the growing complexity of these protocells, they crossed the ill-defined line dividing the living and non-living. Primitive, membrane-bound systems of genetics and metabolism became more like a bacterium than a simple soup of interacting chemicals. The evolving genetic system hit a wall during this upgrade from the original scheme of ribozymes to the new, high-tech proteins. These first mini-genes were 100 bases long, but could no longer grow. Although RNA is complex enough to code information, its capacity is severely limited, making a harder molecule a better long-term store. Reverse transcriptase, an enzyme capable of converting the information in RNA into a DNA molecule, is the protein that introduced this new storage format. Retroviruses, including HIV, utilize this function as a central process in genetic engineering. Reverse transcriptase enables the conversion of RNA genes into a much more robust and stable material, akin to a scribe transferring their work from wax tablets to durable parchment. Modern genes have thousands of bases. The human genome has around 3 billion bases, arranged on 23 paired DNA strands. RNA lost its role as the primary data storage medium within the cell, but it did not become totally subservient. RNA remains the linchpin of life, holding together the different subsystems of a cell. Messenger RNA temporarily copies the genetic information from the DNA library and transports it to the ribosome, which is also based on RNA. The ribosome uses spare amino acids attached to a transfer RNA molecule to churn out the specified protein. The ribosome is a giant ribozyme decorated by attendant proteins, a relic of an archaic world that has taken on critical duties within the cell. The RNA world is far from dead. RNA viruses still vastly outnumber cellular life. In some ways, the interior of a modern cell is the persistence of conditions prevalent in the RNA world, and RNA viruses are merely continuing an ancient way of life. The RNA world's evolving system is theoretical. We can only experimentally demonstrate its existence so far back in time. There is also growing evidence that RNA was not the first self-replicating molecule. It is difficult to explain the synthesis of RNA using prebiotic chemistry. Base formation is simple, created from inorganic hydrogen cyanide from meteorites, but manufacturing ribose is much more difficult. Formaldehyde, also present in meteorites, produces ribose when it reacts with alkaline water. However, this non-specific process produces ribose in low concentrations and is just one of a host of irrelevant sugars. One possible explanation is that abiotic chemistry never created ribose. We've already discussed the hypothesis that metabolism preceded genetics. It's possible that an evolving biochemical network could produce ribose instead of requiring abiotic formation. If this is the case, simpler information-carrying molecules may have emerged first. The polymer TNA, which I discussed in Chapter 1, is one possibility. There may even have been a success of genetic systems, each outcompeting a more primitive rival, until the hegemony of the DNA protein world took over in the first cells, with the previous reigning champion, RNA, clinging on as a go-between. All life on Earth uses a DNA-RNA genetic system, and an array of proteins oversees its metabolism. The last universal common ancestor, Luca, must have possessed these attributes before the archaeal and bacterial domains diverged. The Luca is unlikely to have been a single cell type. It is more likely a community of organisms engaged in a gene exchange organization, a distributed progenitor collective. As different life forms evolved and their cellular systems became idiosyncratic and less flexible, the opportunities for gene sharing lessened. First, the essential machinery for translating proteins solidified, and even now, distant relatives continue to transfer metabolic enzymes. Today, there are two prokaryotic cell types, archaea and bacteria. But it is possible that other, fundamentally different life forms split from the common ancestor, evolved, exchanged genes, and then succumbed to extinction. This might explain the curious presence, in eukaryotes, of some genes that don't seem to resemble anything found in either archaea or bacteria. The first cell's autotrophic or heterotrophic status was a major question. Were they self-reliant, able to extract energy and fix carbon from inorganic reactions, autotrophs, or did they need to consume pre-formed organic molecules from their surroundings, heterotrophs? If the atmosphere was sufficiently reducing at the start, abiotic reactions, such as those caused by UV light, lightning, or black smokers, could have provided the first cells with a ready supply of complex organic substances. As the biosphere grew, this food supply would have gradually dwindled, forcing the hungry cells to evolve methods of generating the substances they needed for themselves, moving from heterotrophy to autotrophy. In a less reducing atmosphere, autotrophy might have developed first, with heterotrophy a later adaptation to living off the efforts of autotrophs. One way of settling the argument is to look at the tree of life, which plots the relatedness of different cells. The root of this tree, the current organism most closely related to all other organisms, would indicate what the universal ancestor was like. Unfortunately, drawing such a wide-ranging family tree is very difficult. The root moves about depending on the assumptions and statistics used. We can't tell whether the Luca was autotrophic or heterotrophic. However, despite their slight differences, the constructed trees share a common pattern. Most show an archaeal hyperthermophile in the middle, suggesting that the mother of all life on Earth lived in hot water. This lends weight to the theory that life emerged in the chimneys of deep-sea vents. But another interpretation is equally plausible. Towards the end of the heavy bombardment, impacts large enough to completely boil away all water and sterilize the Earth had stopped. But smaller impacts, dumping enough energy to heat the oceans substantially, would have continued. The first life may not have been thermophilic, but only cells that could tolerate temperature bottlenecks survived the heavy bombardment and repopulated the world. Consider thermophilic Noah instead of thermophilic Eden. Very few rocks date back to the earliest period of our planet's history. Even if an unbroken geological record existed, the action of prebiotic chemistry would have left no trace. The ambiguity of the first signs of cells in the geological record highlights a central problem in astrobiology. How can we unmistakably identify and distinguish life from similar abiotic processes? To date, we have used three main lines of evidence for the first appearance of terrestrial life. Some argue that Greenland's patch of 3.8 billion-year-old rocks contains the earliest evidence of biological action. Over eons, heat has extensively modified these ancient rocks through metamorphosis. Some researchers claim that the graphite trapped within their mineral grains is rich in the lighter carbon isotope, a trademark of biological processes. Nuclei of isotopes of the same element have different neutron counts. Carbon has several isotopes, including carbon-12 with 6 neutrons and carbon-13 with 7. The environment naturally contains 12C and 13C in a known ratio, but many biological processes favor the lighter isotope due to its easier capture and release from chemical bonds, distorting this ratio. For example, rubisco, the enzyme that fixes carbon dioxide during photosynthesis, produces organic compounds with around 2% more 12C. The researchers claim that just such a change in the isotope ratio is present in the Greenland rock and must have resulted from biological action while the rock was still sedimentary before it underwent metamorphism. This claim is very hotly debated. Its critics point out that certain abiotic reactions also cause distortion of the 12C to 13C ratio, that the rock could not originally have been sedimentary, and that no carbon is present. There is an increasing rejection of such a weakly supported, indirect, and extremely early life date. The second line of evidence for a very early emergence of life rests on a layer of rock in the Warrawoona outcrops in Western Australia. Their supporters claim that these rocks, 3.45 billion years old, contain microscopic features suggestive of cells of 11 species of fossilized bacteria. The size and shape of some of these suggest they may be cyanobacteria, the only group of prokaryotes that can perform oxygen-releasing photosynthesis. This is an extremely troublesome claim, as it predates many other signs of cyanobacteria and evidence for an oxygen-bearing atmosphere by about 1 billion years. The debate rages over whether they are cells, given that purely geological processes like the hydrothermal crystallization of minerals can produce similar forms. The third class of evidence pertains to the dome or column-shaped layered rock formations known as stromatolites. Today, mats of photosynthetic and heterotrophic bacteria living in warm, shallow water create such structures. The cells excrete mucus, which traps drifting sediment and binds it into a covering layer. The bacteria recolonize the new top surface open to the sun, and over time, these biofilms build into large structures of layered sedimentary rock, as seen in Figure 8. At this very early date, researchers found similar-looking structures in 3.5 billion-year-old rock in the Warrawoona area, citing them as evidence of bacterial action, including photosynthetic cyanobacteria. These ancient structures lack cell fossils, and the curved layers could have been produced by abiotic sediment deformation. One of the first signs of life was stromatolites, along with other evidence. The debate is about how soon biology can identify them. Although the exact timing of life's emergence is controversial, the evidence is overwhelming, dating back about 3 billion years. Prokaryotes were widespread at this point. They drifted near the ocean's surface and along the edges of coastlines, inhabiting deep and shallow hydrothermal habitats. They formed ecosystems with photosynthesizing autotrophs and grazing heterotrophs. Considered photosynthesis, but it has played a pivotal role in the Earth's evolutionary history, and it is time to return to it in detail. Autotrophy lets cells get the energy or carbon they need from organic molecules, but chemosynthesis still keeps life in small areas with strong redox gradients, like black smokers or hydrothermal pools. The development of photosynthesis, surviving on light, liberated cells from these locations and left them free to colonize the whole planet. The chlorophylls, which form the basis of photosynthesis, are believed to have originally evolved to protect the proteins and DNA of early cells against the harsh UV radiation streaming down onto the surface. Currently, sunlight produces the ozone layer, which absorbs UV from the oxygen in the atmosphere. However, 3.5 billion years ago, the atmosphere was nearly oxygen-free, offering little protection from UV radiation. Therefore, surface life evolved molecules capable of absorbing this light. Once they have absorbed a particle of light, they must dissipate its energy before absorbing more. One way to accomplish this is to pass excited electrons onto neighboring proteins. The first photosynthetic organisms used this waste electron energy to fix carbon and drive useful redox reactions. However, there is a constraint to the derivation of energy in this way. The cell needs a steady supply of replacement electrons, meaning it must be near a source of reducing power. Some bacteria gain these electrons by oxidizing hydrogen sulfide, a chemical abundant in hydrothermal Mao pools. However, to be fully autonomous, a cell must cut this final tie. Nature's solution was to take electrons from water molecules. Thus, photosynthesis could provide a renewable energy source and release life from its dependence on reduced environmental ions. This development necessitated a new, supplementary photosystem, which requires much more power to split water, but provides enormous amounts of energy for the cell. This advanced form of photosynthesis creates pollution, causing one of the greatest ecological disruptions in the history of life. Water splitting produces an extremely poisonous gas that the photosynthesizing cells excrete into the air. This toxic chemical forms highly reactive products, such as peroxide, that ravage life's complex organic molecules. It is oxygen, and a vast amount of it in today's atmosphere comes directly from photosynthesis. The only organisms capable of splitting water in this way are cyanobacteria and the plant cells that formed an endosymbiotic relationship with them. Oxygenic photosynthesis could have evolved as early as 3.5 billion years ago, but the first evidence of rising oxygen levels in the atmosphere only appeared 2.5 billion years ago. Oxidation steadily increased in the atmosphere and water, causing the exposed reduced minerals to oxidize. Rocks from that period show thick bands of deposited iron oxide, and the planet's surface was rusting. Once the Earth's crust had soaked up all the oxygen it could, its atmospheric levels started to rise rapidly. Previously, life was anaerobic, allowing it to run its metabolism without using oxygen as a final electron acceptor. In fact, much of life still finds even trace amounts of oxygen extremely toxic, and confines itself to places like deep water or underground, where it cannot reach. Aerobic life survives because it develops mechanisms to cope with oxygen's destructive effects. By 2.2 billion years ago, the atmospheric concentration of oxygen had climbed to about 1% of its present value, and suddenly something happened to life on Earth. Previously, cells, including the photosynthetic cells that were generating it in the first place, had limited the damage that the products of oxygen reactions could wreak. One method is to produce scavenger molecules that combine with and neutralize the reactive oxygen chemicals before they can attack crucial polymers. However, some organisms took a new tack and utilized this immense oxidizing power. As I described in the section on respiration in Chapter 1, oxygen makes a fantastic final electron acceptor at the bottom of the transport chain. Aerobic cells could extract far more energy from organic molecules by breaking them down further and exploiting oxygen. The rising atmospheric oxygen levels had another important effect on surface life. Solar UV light splits apart oxygen molecules high in the atmosphere, producing ozone, which is a strong UV absorber. So, as long as there is an appreciable presence of oxygen in the air, sunlight naturally builds up a strong ozone shield, limiting the amount of UV radiation that can reach the ground. Scientists estimate that DNA-damaging UV radiation on early Earth was more than 40 times more intense than it is today. One cannot overstate the importance of photosynthesis. High concentrations of reduced inorganic compounds are a relatively limited resource, which probably constrained the spread of early life. The evolution of photosynthesis provided light-harvesting cells and the ecosystems they supported with an effectively unlimited energy source. The oxygenation process also produced oxygen, allowing for more efficient burning of nutrients and creating an ozone shield against UV radiation. Photosynthesis allows cells to rebuild themselves using only carbon dioxide, water, and sunlight energy. These simple building blocks are likely common on other terrestrial planets, so photosynthetic systems could provide metabolic energy to alien ecosystems across the galaxy. Extraterrestrials synthesize porphyrins, the basis of chlorophyll, hemoglobin, and similar metal-containing molecules, suggesting a widespread presence of chlorophyll in alien worlds. In later chapters, we'll look at the plausibility of harvesting the light of other stars and how astronomers might detect the telltale signs of photosynthesis from light years away. The earliest eukaryotes appeared around 2 billion years ago, but exact dating is difficult because the first cells were as small as archaea or bacteria and had no cell wall to leave a recognizable fossil. Their evolution intriguingly corresponds with rising oxygen levels. The need to protect the fragile DNA from oxidation may have driven the nucleus's evolution. This is no coincidence. During this period, dissolved nutrient levels were also low. Endosymbiosis, which involves ingesting mitochondria and chloroplasts as organelles, may have improved energy efficiency and recycling within previously free-living organisms. Many eukaryotes, such as our human cells, are aerobic. An oxygen-laden atmosphere is crucial to their existence. In this sense, oxygen is a double-edged sword for complex eukaryotes. They cannot survive without it, but it can also powerfully destroy if the cells do not handle it properly. Mitochondria, once believed to be free-living bacteria, carry out many risky energetic reactions within the developing eukaryotic cell. Cyanobacteria, the only prokaryotic group capable of oxygenic photosynthesis, is the ancestor of chloroplasts. In addition to their sophisticated interior organization, improved DNA capacity, and genetic control, eukaryotes have one other enormous advantage over prokaryotes, an evolutionary leap they made about 1 billion years ago. Eukaryotes are exceptionally adept at getting together, building much larger organisms from single-cell congregations. The adult human body, for example, is a colossal colony of some 100 million cells, sacrificing their individuality to the health of the whole organism. There are over 200 specialized types of cells, each expressing a subset of the body's complement of several 100,000 different proteins. And this exquisite complexity self-organizes from a single fertilized egg cell. While fungi and microscopic protists have independently evolved multicellularity, animals and plants are particularly adept at it. Although prokaryotes often stick together into microbial mats, such as stromatolites or the plaque that forms on your teeth, true multicellularity is distinct from simple colonial living. Even in a primitive animal, cells differentiate into a massive diversity, each with a specific function, anchored onto a sturdy extracellular matrix, communicating with each other and ceding their reproductive rights to a few germline cells. A few examples of multicellular organisms exist in the bacterial world but are comparatively simple and temporary. Myxobacteria, for example, release a signal when nutrients run out and swarm together into a structured slime, producing a single upright stalk of cooperating bacteria that eventually releases a puff of reproductive spores. However, there are no bacterial seaweed or archaeal jellyfish fronds on Earth. The lack of widespread multicellularity among prokaryotes is surprising, considering its clear advantages. Larger organisms are safer from predators, more effective at nutrient uptake and storage, can better regulate their internal environment, and the division of labor can make the whole organism more versatile. Prokaryotes may lack communication sophistication or the required genetic control. However, they remain the most successful organisms on the planet, perfectly adapted to rapid reproduction and capable of exploiting various energy sources. Some genetic evidence dates the earliest complex animals to around 750 million years ago, but the first fossils only appeared about 600 million years ago. Complexity demanded several crucial developments, including the protein collagen, a component of the extracellular matrix that holds animal cells together, and homeobox genes, a set of switches that establish the features of a body plan. Around 540 million years ago, the oxygen level in the atmosphere was about 10% of its current value. This date coincides with another revolution for life on Earth, when, quite suddenly, a spectacular diversification of marine animals appears in the fossil record. Every basic body form we see today, such as mollusks, like mussels and snails, arthropods, like lobsters and insects, and chordates, like humans and fish, first appeared in this period. The fossils also document many weird and wonderful forms that have not survived to the present day, such as the armored predator with a pair of jagged tentacles and rows of swimming fins. The Cambrian explosion, an apparent burst of evolutionary innovation, has sparked intense debate over its origin. Some scientists argue that it does not represent a sudden biological event, but rather that fossilization conditions changed, and only then were pre-existing species preserved. Others claim that a specific incident sparked an evolutionary race between competing organisms. For example, the evolution of the eye would have allowed for active hunting, triggering an arms race of adaptations in predators and prey. Another explanation is particularly relevant to astrobiology and the implications for planetary climate stability. We described how planets within their star's habitable zone tread a thin line between opposing disasters. Rising temperatures can set off a runaway greenhouse effect, while runaway glaciation results in plummeting temperatures and a global covering of ice. Earth's current climate seems unusually cool. For 90% of its history, there was no ice cap on the poles. However, records of previous climates, held in rocks, also show that deep glaciation befell the planet on several occasions between 750 and 600 million years ago. Not only did these episodes collapse the surface ecosystems, but they also came close to wiping out eukaryotes altogether. Once the cooling started, the growing regions of white ice reflected more and more of the sun's warmth, creating ever colder conditions. The polar ice caps expanded until they almost touched the equator and may even have blanketed the entire surface in a thick ice sheet. This snowball Earth sealed its seas, obstructing sunlight and precipitously reducing photosynthesis. Deprived of its source of oxygen, the seas turned anoxic for the first time in over 1 billion years. The Earth effectively suffocated itself, and many organisms would have gone extinct. We are still here, which implies that some prokaryotes and our eukaryotic ancestors clung on, surviving in havens of meltwater or thin ice on the supercontinent, or holes in the glaciers around volcanic islands. Some researchers are dubious that the surface ever completely froze and argue for more of a slush ball Earth with pockets of clear water. Either way, these epic ice ages lasted several million years and ended only by the saving grace of our planet's active volcanism. With silicate rock erosion almost non-existent, carbon dioxide accumulates in the air to as much as 350 times its current level. Finally, a well-insulated atmosphere's powerful greenhouse effect began to thaw out the planet. During this recovery period, the weather would have been absolutely frenzied as warm air from the tropics expanded into the still frozen wastelands around the poles. Giant ripples in sedimentary rocks from this time suggest long-running storms assaulted the seas with wind speeds of over 70 kilometers per hour. The climate eventually settled into equilibrium, and life repopulated its former niches. The theory is that the release from such a harsh environment provided the evolutionary prompt for complex animals, leading to the Cambrian explosion. As this journey through time approaches the modern day, we see multicellular plants and animals joining the prokaryotes in colonizing the land, the once barren continents flushing green with vegetation, as organisms evolved flattened leaves, rigid wood, flowers, and fruit. Animals similarly developed their adaptations, including yolky eggs, flight, warm-blooded physiology, and now self-conscious intelligence as the continents slide into familiar positions. Extinction is as much a natural part of evolution as innovation and the emergence of new forms.
The Rocky Mountains, the backbone of North America.
Yeah.